Hello everyone. So today we are going to discuss about the NEET 2022 biology paper, in which we will be discussing the entire 90 questions. All right. So let's start with the very first question. And this says that which of the following is not a method of ex situ conservation? Now, following of options has been given, and this is asking about the method that is not indicative of ex situ conservation. Now, let me tell you that there are two methods basically of conservation in biodiversity. So two methods of conservation of biodiversity. So two methods have been proposed. So these two methods are the in situ method. The first one is the in situ method of conservation, and the other one is the ex situ method of conservation. All right. Now, in situ method of conservation basically relies upon the fact that this is the on-site conservation of the species. All right. So this is basically the on-site conservation of plants and animal species, or species you can say. So on-site conservation of species, that can either be animals or plants. All right. While ex situ conservation is defined as the off-site conservation. Off-site conservation means what? Off-site conservation basically means that the organism or that particular species will be taken out from its natural habitat and should be cultivated or should be grown or should be nurtured in a environment which is not its natural habitat, but it will be provided with all the amenities that are found in its own natural habitat. So that's why it is called as off-site conservation. That means not the natural habitat, but the conditions that will be provided to that particular organism to survive, that will include all the natural amenities that the organism was availing in its natural habitat. So that's what this is called as off-site conservation. All right. Now, the examples of in situ conservation, since this is the on-site conservation and the species will be protected in its own natural habitat. So basically all the examples include like national parks, then wildlife sanctuaries, then there is biosphere reserves, then wetlands or swamps, you can say. All right, so national parks, wildlife sanctuaries, biosphere reserves, then wetlands, then sacred groves, etc. These are the basically examples. All right. While ex situ, the examples of ex situ conservation, it primarily includes, pollen bank, gene bank, seed bank. So pollen or gene or seed banks. This is in case of plants. All right. Then in case of animals, if we see, then there are zoological parks, which are also known as zoos. Then there is botanical gardens, which are being constructed and built up. So botanical gardens. Then there is, method of micropropagation. Micropropagation is basically a method of tissue culture technique which is performed under lab conditions. So micropropagation, also called as tissue culture. This is also considered to be tissue culture. Then cryopreservation technique, where the, you know, any living tissue that can either be obtained from the animal or can be obtained from the plant. Any living tissue is actually preserved at a temperature of minus 196 degrees Celsius, and that too in liquid nitrogen. All right, so this is the storage or preservation at minus 196 degrees Celsius, and this basically belongs to liquid nitrogen. So this is also offsite conservation. All right. Apart from this, other methods also includes, methods of like, in case of this offsite conservation or ex situ conservation, this also includes in vitro fertilization, which is also considered to be as IVF. All right. So let us take a look into the following option, which is not a method of ex situ conservation. So we have seen that this one, there is the option A is the in vitro fertilization. Again, this is an ex situ method of conservation. All right. So this is not an option. Then national parks, definitely this is not a method of ex situ conservation because this belongs to in situ method of conservation. So that's why the correct answer will be here only. But let us take a look into the other options. Micropropagation and cryopreservation. So micropropagation and cryopreservation both belongs to ex situ conservation. So that's why the correct answer is option number B over here. Clear? Now, question number second. Now, question number second says that you have to identify the correct set of the statements. All right, you have to choose out the correct set of the statements out of the following statements that has been given. All right, now let us take a look into the statements first. So the first statement says that the leaflets are modified into pointed hard thorns in citrus and bougainvillea. All right. So the leaflets are modified into pointed hard thorns in the citrus and bougainvillea. So is that so? Is that what we have studied also? So let us take a look in the other options also, and then we'll move on to this statement. Clear? Now, the second statement says that axillary buds forms slender and spirally coiled tendrils in cucumber and pumpkin. So this is definitely a correct statement, because in case of cucumber and pumpkin, if we see, then the axillary buds basically, they are responsible for forming the slender and spirally coiled tendrils, which are the supporting structures. So in option B, if you see, then axillary buds, they form tendril. So forms tendrils, and these tendrils, these are basically the supportive structures. These are considered to be a supportive structure. So which are supportive in nature. They help in climbing basically. Supportive in nature or helps in climbing. Helps in climb. Clear? So this is a correct statement actually. Now the second statement, option C, says that stem is flattened and fleshy in Opuntia and modified to form the function of leaves. So again, this is also a true statement because Opuntia is a kind of a xerophytic plant body. And in case of this Opuntia, the stem becomes flattened and its stems only performs the function of leaves. So definitely this is also a correct statement. All right, because Opuntia is actually having a xerophytic adaptation. So it is a xerophyte where its stem actually gets modified and turns, and it actually performs the function of leaves, where its stems becomes flattened and fleshy. where its stem becomes flatter and fleshy to perform the function of leaf. The functions of leaves. True. That means the photosynthesis process. So this is also correct statement. Now option D says that rhizophora shows vertically upward growing roots that helps to get oxygen for respiration. Again, this is a correct statement because let me tell you that rhizophora, they usually have pneumatophores. All right, rhizophora is one such plant body, whose roots are actually modified, and its roots are modified in the sense that the roots actually comes upward from the soil. It grows upward, from the soil, and they have a very unique structure present in their roots, which are called pneumatophores, through which they perform the process of respiration. So definitely this is also correct statement because rhizophora have pneumatophores. So they have pneumatophores that helps in root respiration. So that helps in root respiration. Now the last statement says that subaerially growing stems in the grasses and the strawberry that helps in the vegetative propagation. Now the subaerial stems, which usually grows in case of grasses and strawberries, they help in vegetative propagation. So this is again also a correct statement because if you remember, then grasses and strawberries, they are the example of subaerial stems only. That means these are the structures, or these are the plant body that grows partially in attachment to the soil and partially in air. So that's why they are an example of subaerial growing stems. So this is also correct statement. So you can say that grasses and strawberry are examples of just a second. Examples of subaerial stems. So definitely they will be used for vegetative propagation because grasses is one of those plant bodies that propagates very fast through vegetatively. All right. So this is also correct statement. Now, the last statement which is left over here, so all these four statements are correct. Now, the last statement which is left over here is the statement number 1, which is the leaflets are modified. Since we have left this statement for reviewing, for the purpose of reviewing, so let us take a look. So the leaflets are modified into pointed hard thorns in case of the citrus and bougainvillea. Now let me tell you that in case of the citrus and bougainvillea, the axillary buds of the stems, they are modified. They get modified, in the form of those hard pointed structures, which actually helps these plants from the grazing animals or browsing animals, prevent these plants from being grazed by the browsing or grazing animals. All right, so that's why this statement is actually incorrect over here, because it is not the leaflet, but it is actually the axillary buds. All right, so A is basically the incorrect statement. So you can say that the axillary buds of stems, gets modified into, so this actually gets modified into hard, woody and pointed thorns. So hard, woody and pointed thorns that helps these plants, that means in citrus and bougainvillea. In citrus and bougainvillea, that helps these plants from being prevented by grazing animals. So that helps these plants being browsed by or being grazed by browsing animals. All right, so this is the incorrect statement. So out of all the 5 statements, only the option A is the incorrect statement and all other statements are correct. So let us take a look into the options. In case of the options that you can see over here, C option is the correct option because here B, C, D and E only are the correct one. All right, so that means the correct answer is option number C. Now, let us move into next question, which is question number 3. Now, question number 3 says that given below are the 2 statements. That means decomposition is a process which says that decomposition is a process in which the detritus is degraded into simpler substances by the process of microbes. Now, definitely this is a correct statement. Now, basically 2 statements are given, and in the light of the above statements, you have to choose the correct answer from the options given below. All right. Now, the statement 1 says that decomposition is a process in which the detritus is degraded into simpler substances by microbes. Now, statement 1 is basically considered to be correct, all right, because the definition of, the definition of decomposition is true. Statement 1 is correct because decomposition is a process. Decomposition is a process of breaking down of complex organic matter, or you can say breaking down of the detritus. So detritus is the organic matter only. So breaking down of detritus, which is actually considered to be as the raw material of decomposition. So raw material, all right. Detritus or complex organic matter into simple forms or simple substances by microorganisms, in which the major microorganisms which are involved are the bacteria and the fungi. So this is a correct statement. Now let us take a look into the second statement. So second statement says that decomposition is faster if the detritus is rich in the lignin and chitin. Now let me tell you that decomposition, there are various factors that actually affects the process of decomposition, and one of the factors that affects the process of decomposition is basically the composition of detritus. That matters very much. All right, so if the composition of detritus is rich in lignin and chitin, then the decomposition is considered to be slower. Why? Because these lignin and chitin, they are the complex polymers. All right, so they will definitely be taking, large amount of time, or more amount of time for its decomposition. But those detritus which are rich in nitrogen and sugars, they are being decomposed by the microorganisms very fastly. All right, so we can technically say that this statement is actually considered to be false. So statement second is actually incorrect, and the reason for this is incorrect because the composition is faster if the detritus is rich in nitrogen and water soluble organic substances, or you can say simply directly you can say sugars. So if the detritus is rich in nitrogen and sugars, however, it is slow, it is slow if it is rich in lignin and chitin, which are actually the complex polysaccharides. So lignin and chitin. And why it's slow? Because this lignin and chitin, these are complex polysaccharides, which usually takes time for decomposition. Complex polysaccharides. So basically, we can say that this process of decomposition is not only affected by the composition of the detritus, but also by various other climatic factors as well. But since here it has been asked only regarding the composition, so we will be looking into that part of it. All right. So let us take a look. That means what is the statement? So both statement 1 and statement 2 are correct. Again, this is wrong. Both statement 1 and statement 2 are incorrect. Wrong. Statement 1 is correct, but statement 2 is incorrect. This is the correct answer, correct option. Statement 1 is incorrect, statement 2 is correct. Again, this is wrong. That means option number C is going to be the correct answer. Clear? Now, let's move ahead with question number 4. Now, question number 4 says that hydrocolloid carrageen is obtained from. Now, this is a very direct question, and that has been asked from the ecological importance of the algae in the entire biosphere, if we talk about. And let me tell you that what is actually hydrocolloids. So hydrocolloids are actually the water holding substances. All right. So hydrocolloids are the water holding substances. That is, generally obtained. So since these are the water holding substances, so these hydrocolloids, these are generally obtained from so these hydrocolloids, these are actually the water holding substances that we usually obtain from two categories of algae, and that is your brown algae and red algae. All right. So that means these are the water holding substances that we obtain from, and these hydrocolloids, they are actually being commercialized in the market. So they are very commercially successful. All right. They are being commercially used. So that we obtain from algin, and algin is a category of brown algae. Let me tell you. So algin is basically alginic acid is found in the cell wall of the brown alga. So that's why. So this is obtained from brown algae, algin and carrageen. Carrageen, which is a category of red algae. So that means from both of them, this is obtained. That is used commercially. That is used commercially. So in biological terms, this brown algae is considered to be as phaeophyce. So this is also known as phaeophyce. while this carrageen is also considered to be as, or this red algae we can say rather, red algae, this one is known as rhodophyceae. Rhodophyceae. So that's why the correct answer will be both, including both the options. So chlorophyceae and phaeophyceae, no. Phaeophyceae and rhodophyceae, yes. Rhodophyceae only, no. Phaeophyceae only, no. So that's why the correct answer is option number B over here. Clear? Now, let's move on to the next question, which is question number 5th. Now, question number 5th says that which one of the following plants shows vexillary aestivation and along with diadelphous stamens. All right. So vexillary aestivation and diadelphous stamens. Now, let me tell you that this vexillary aestivation is basically considered to be as one such kind of an aestivation, which is also considered to be papilionaceous. All right. So vexillary aestivation, and it is talking about, and diadelphous. Diadelphous condition means basically the stamens are arranged in two whorls. 9+1 kind of an arrangement is there. So that is called diadelphous condition. So vexillary aestivation, which is also known as papilionaceous. Naceous aestivation. So how the aestivation is actually over there? So there is a larger leaf like this, larger petal, then beneath there's a single large one petal, then beneath this one large petal, the smaller two petals are there, and then inside these smaller two petals, the other two smaller petals are there, which are joined to one another like this. So this is considered to be as vexillary aestivation, like this one. All right. So total 5 are there, but the vexillary, in case of the vexillary aestivation, how the arrangement is there? It is 1 plus 2 plus 2 kind of arrangement is there. 1 plus 2 plus 2. So this is basically, aestivation is basically a process of the arrangement of petal or sepals on floral axis, how they are actually arranged. All right. So that is the case. So this is basically 1, then this is 2, and then this is 2, if you can see over here. So this is the vexillary aestivation, and this is one such unique aestivation which is usually found in case of all the leguminous members, or leguminous plants categories. It is being found. All right, and it's a characteristic feature of Fabaceae family, Fabaceae or Leguminosae, if I say. All right. So this is a characteristic feature of feature of Fabaceae family, which is also considered to be as the Leguminosae family. All right. And what is diadelphous condition? Now let me tell you about diadelphous condition. Is basically a condition where the stamens are arranged in two whorls. So diadelphous. And in case of the Fabaceae, Fabacean members, if I tell you, then this is again, this is also, this diadelphous condition is also a characteristic feature in case of Fabaceae, because it is usually arranged in a pattern of 9+1, which indicates that 9 stamens in one whorl and the 1 stamen in the other whorl. So 9 stamen grouped in one whorl. one word. And this is one single stem in another word. Another word. Layer or word. So that's why it's diadenphous condition. All right, so this is also a characteristic feature of Fabaceae. So out of all the options that we can see, so Colchicum autumnale. Now Colchicum autumnale basically belongs to your this one Liliaceae family. Colchicum also and this Allium cepa also. Both these members, they basically belong to, so Colchicum and Allium cepa. They both belong to Liliaceae family. Belongs to Liliaceae. So definitely this is not an option. All right. And the last which is left is the, I guess, Solanum nigrum. Now Solanum nigrum, which is also considered to be as Makoi plant, this belongs to Solanaceae family. So Solanum nigrum, which is called as Makoi. So this basically belongs to Solanaceae family. So the only option left is the Pisum sativum over here. That actually belongs to Fabaceae. So here will be Pisum sativum, which is pea plant, garden pea plant. You can say. So the correct option will be option number B over here. All right. Now let's move on to next question, which is question number 6. Now, the next question says that which of the following is incorrectly matched. So you have to find out the incorrectly matched statement. All right. So with the current given options. So the first option is ectocarpus and it is related with fucoxanthin. Now let me tell you that ectocarpus is basically a category or it's an example of brown alga. All right. So ectocarpus is a brown alga and this has the presence, and since this has been written over here, fucoxanthin, so this is actually, fucoxanthin is basically the pigment, all right, that is found in case of the brown alga only, fucoxanthin. So definitely fucoxanthin is the main pigment, you can say. Fucoxanthin, a main photosynthetic pigment in brown alga. So this is actually a correct statement. A main photosynthetic pigment. Clear? Apart from chlorophyll A and C. Apart from chlorophyll A and chlorophyll C. All right. So this is basically correct. All right. Now, option B says Eulothrix. Eulothrix, and that is related to mannitol. Now, let me tell you that Eulothrix is a category of green alga. It's a member of green alga. All right. That means chlorophycean member. So, Eulothrix basically belongs to green alga, and green alga usually have starch as their reserve food material. So starch as reserve food material. All right. So here the reserve food material mentioned over here is mannitol. All right. And mannitol is the reserve food material for brown alga. So mannitol is reserve food for brown alga. So definitely this is actually the incorrect statement, all right, or incorrect option matching, you can say. So we have actually found out, but let us take a look into the other options also. Porphyra, there is Floridian starch. Now porphyra is a red alga. So porphyra, this is a category of red alga, and this has Floridian starch as reserve food material. So this is true. So Floridian starch as reserve food. So this is again just true. Then the last option is volvox, and it is starch. Again, volvox is a colonial green alga. So this is also an example of green alga. So definitely this is also considered to be as the correct statement. So this is also the correct statement. That means, rather we can write over here. So starch is a food. All right. Now, so this is also correct. So since we have to find out the incorrect matching out of the all the four options, so the incorrect matching is option number B over here. So the correct answer is option number B. All right. Now let's move on to next question, which is question number 7. Now question number 7 says that the process of translation of mRNA to proteins begins as soon as. Now, there are certain statements that regarding the translation process has been given. Now, we have to find out the correct statement out of all the statements. So now and since this is talking about the beginning of the process only, so we have to focus on that part also. So the process of translation of mRNA to proteins, it begins as soon as the small subunit of ribosome encounters the mRNA. Small subunit basically means either it can be in the form of prokaryotic ribosome or it can be in the form of eukaryotic ribosome. The smaller subunit has to be taken. All right. Then the larger subunit of ribosome encounters the mRNA. Then both the subunits join together to bind with the mRNA. And then the tRNA is activated and the larger subunit of ribosome encounters the mRNA. All right. Now, let me tell you that as soon the process of translation, if mRNA is needed to be translated into proteins, then the first important process is that when this smaller subunit of ribosome, all right, when the smaller subunit of ribosome that will be encountering the mRNA, then only the process of translation will start or it will begin, because after binding of the smaller subunit with the mRNA, there will be the binding of the bigger subunit, and then only the tRNA, the activation of tRNA, and all those things will be started upon. So we can say that once the small subunit, once the small subunit that can either be, if it is prokaryotic ribosome, so it is usually 30S, or and if it is eukaryotic ribosome, then it is usually 40S. Clear? 30 or 40S encounters mRNA, encounters the mRNA. The process of translation, the process of translation of mRNA into proteins starts. Into proteins starts or begins, because this process is then followed by this process is then followed by binding of bigger subunit, binding of bigger ribosomal subunit. Ribosomal subunit, and when both the subunits they bind together and they are joined, then this results into activation of the tRNA after the attachment. All right. So then tRNA is activated. So then this tRNA ultimately gets activated by addition of amino acid, which is considered to be as the amino acylation of tRNA prior to the attachment of, this is prior to the attachment of ribosome. All right. So that's why the correct answer will be option number 1. Because once the small subunit will be joining, then only the larger subunit of the ribosome will come and join it. Both will be forming a complex, and then there will be the activation of the tRNA. All right. So this is how the process is carried out. So let's talk about the next question, which is question number 8. Now, question number 8 says that you have to match the column with the following list. All right, that has been given below over here. Okay, so these are some of the elements that has been given. You can consider it to be as the macro element or micro element. So regarding with their functions. All right. So if we just deal with manganese, so the first option is manganese over here. So manganese, let me tell you that manganese is one such element which is related with the splitting of water. That means photolysis of water during the process of photosynthesis. So option A, which is manganese, which is shortly termed as Mn, this is related to photolysis of water. That means splitting of water molecule in the presence of, this one. That means your calcium ions also. So this is related to photolysis of water during the process of photosynthesis. All right. So that means this is going to match up with option A over here. Then option B is with manganese. Magnesium. Magnesium is basically required, or that is usually related to your, that this is usually related to the activation of the enzymes in the respiration process, because this magnesium, it acts as a cofactor. This basically acts as a cofactor in many of the enzymatic reaction, and specifically in the process of respiration. All right. So that's why magnesium is involved in, or involved in activating enzymes, for respiration. All right. Now, the C option is boron. Now, boron is one such microelement which is related with pollen germination. Okay, so this is with B option and this is going to be with C option. So boron is related to required for pollen germination. All right. And the last option is option D, which is iron. And iron, we all know that it is primarily responsible. It is responsible for activating the enzyme catalase. All right. And this catalase is responsible for degrading the hydrogen peroxide or converting hydrogen peroxide into water and oxygen. So this is with option D over here. That means iron. Just a second. So iron. So iron is related with activation of enzyme catalase. Enzyme catalase. All right. Now let us take a look into the matchings. Now you have to choose the correct answer from the options given below. So A is going to match up with fourth option. A is going to match up with fourth option. B is going to match up with third option. B is going to match up with third option here. Then C is going to match up with second option, this one. And D is going to match up with first option, which is this one. So our correct answer is option number D over here. All right. Now, question number 9. Now, question number 9 says that which one of the following never occurs during the process of mitotic cell division? All right. Now, this question says that which is the following event that never occurs during the process of mitotic cell division? So, spindle fibers attaches to kinetochores of the chromosome. Again, this is all. This definitely takes place during the process of mitosis, because until and unless these spindle fibers are attached to the kinetochores of the chromosome, that means the protein component of the centromeres of the chromosome, they won't be stabilized into their own positions. So this is actually correct. This happens in case of, this is correct. Then movement of centrioles towards the opposite poles. Definitely this also occurs, movement of centrioles towards the opposite poles. All right. So this is also a correct statement that happens up. Then pairing of homologous chromosomes, and then coiling and condensation of the chromatids. All right. Now, pairing of homologous chromosome is an event of meiosis, but not mitosis, let me tell you, because pairing of homologous chromosome is considered to be as a phase or a characteristic feature of the zygotine of the meiosis 1. All right. So let me tell you that this option, that means this one, is considered to be actually incorrect, because pairing of homologous chromosomes is related to zygotine phase of meiosis 1. So this is basically a characteristic feature of zygotine phase of meiosis 1. That means this pairing of homologous chromosome, once they are paired, they result into bivalent or tetrad formation. So they result into bivalent formation. That means one chromosome will be coming from the mother and the other chromosome will be coming from the father. So that's why called bivalent. Altogether considered to be as tetrad. That is tetrad. Clear? So this is a phase which is related to the zygotine phase, while Coiling and condensation of chromatids, this happens in case of the prophase only, prophase of the mitosis, early prophase, because in case of the early prophase, the chromatids are present, but those chromatids in the late prophase, they get converted into fully condensed chromosomes actually. All right. And these are actually the events, spindle fibers attaching to kinetochore. These are one such events that basically occurs in both mitosis and meiosis. So we cannot segregate it. So this is also the correct statement, you can say. All right. So the event, this is, since this is the event pairing of homologous chromosomes, this is an event of meiosis 1. So that's why this is the correct answer option for this question. Option C. All right. Now, let's move on to the next question, which is question number 10. Now, question number 10 says that given below are the two statements. So, two statements are actually given over here. All right. And statement 1 and then statement 2. And you have to identify, that means out of the two statements, that in the light of the above two statements, you have to choose the correct answer from the options given below. All right. Now, let us take a look into the statement 1. Now, statement 1 says that the primary CO2 acceptor in the C4 plants is phosphoenol pyruvate, and it is found in the mesophyll cells. All right. So, let me tell you that the statement 1, which says that the primary CO2 acceptor of the CO2, the primary CO2 acceptor in the C4 plants is phosphoenol pyruvate. Definitely, this is a correct statement, because let me tell you that C4 plants usually have two cells. One is called as the bundle sheath cells, and the other is called as the mesophyll cells. All right. And these C4 plants, the first and foremost acceptor of the CO2 in case of the C4 plants is phosphoenol pyruvate only. All right. So the CO2 is basically accepted by phosphoenol pyruvate in the presence of enzyme peptase, which is phosphoenol pyruvate carboxylase, carboxylase enzyme. All right, in the peptase enzyme, and this ultimately gives rise to a 4-carbon molecule. All right, or a 4-carbon product, C4 product, which is either considered to be as oxaloacetic acid, that can either be oxaloacetic acid or aspartic acid or malic acid. It can either be of them, or malic acid. Clear? Now this happens, this entire reaction, this happens up in case of mesophyll cells of C4 plants. In mesophyll cells of C4 plants. So this is a correct statement, definitely. All right, so this is correct. Clear? Now the second statement says that mesophyll cells of C4 plants, they lack Rubisco enzyme. All right, so definitely this is, if you just look into, since the product has already been formed, so let me tell you now the statement 2, which says that the mesophyll cells lack the this one, Rubisco enzyme. So this is again a correct statement, because this Rubisco enzyme is found in the bundle sheath cells of the C4 plants, but not in the mesophyll cells. So we can say that Rubisco enzyme is found in bundle sheath cells. Bundle sheath cells, which is only and only found in case of C4 plants, not in C3 plants or in C2 plants. So in bundle sheath cells of C4 plants and not in mesophyll cells. So that's why this statement also is the correct statement. So with this, we can conclude that this is also a correct statement. So this indicates that both the statements are correct. So this means that both statement 1 and statement 2 are correct over here. All right. So this is the case. That means option A is the correct answer. So let's talk about the next question, which is question number 11. All right. Now this question says that which one of the following is not true regarding the release of, energy during the process of ATP synthesis, you can say, through the process of chemiosmosis. All right. So it basically involves, so basically some of the features of the chemiosmosis process has been given. Now let me tell you that what is chemiosmosis. So chemiosmosis is one such process that results into production of ATP. All right, and there are some of the main ingredients, you can say, or main requirements, you can say, that is involved in the process for the production of ATP. All right, so we can just note down this point that chemiosmosis is a process of ATP production. So basically, this theory was given by Peter Michel. All right. So Peter Michel was the scientist who gave this concept of chemiosmosis, through which this ATP can be produced. Now, for this production of ATP, for this production of ATP, certain requirements are there, and so this therefore chemiosmosis actually requires chemiosmosis requires. So the requirement that is in the process of ATP production are: first, there has to be a membrane, and that membrane, all right. So this membrane depends that this membrane, whether the process is taking place inside the mitochondria or inside the chloroplast. So a membrane is required, all right. Then a very important protein that will produce ATP, that means complex 5, which is ATP synthase. Complex 5, which is ATP synthase, because this is the only complex which is going to harness ATP. All right, that will be producing ATP. Then there has to be a proton pump. There has to be a proton pump. So this proton pump is actually present inside this ATP synthase only, which actually creates a gradient for ATP synthesis. And there has to be, along with the proton pump, there has to be a gradient developed for the proton. So we can say a proton gradient. All right. So these are the requirements for ATP synthesis or ATP production. All right. So basically, if we just look into, or if we just, you can say, consider all the statements and all the options. Now, let me tell you, before considering all the statement, now let me tell you that, this membrane is basically responsible for holding the ATP synthase. This basically holds up the ATP synthase, through which the ATP will be synthesized. Now, energy is basically used. All right, a little amount of energy is already being used for pumping the protons across the membrane, that actually creates a proton gradient. So we can say that proton gradient is basically created as a result of proton pumping. So proton gradient is created due to proton pump. So this gradient is basically created, this gradient is basically created due to the proton pump, and this gradient, let me tell you, is basically developed within the thylakoid lumen. That means proton gradient basically means high concentration of protons, high concentration of protons in thylakoid lumen, in the lumen of the thylakoid, not in the stromal region. All right, because stroma will be having a greater concentration of the proton, so the proton will be pumped from the stroma to the lumen. All right, so that is the case. And apart from that, there is a very important enzyme also that is required in this entire process. And that enzyme is considered to be as the NADP reductase enzyme. So NADP reductase enzyme. Now, this enzyme is basically located on the stroma of the chloroplast, or on the stromal side of the chloroplast, you can say, of the membrane. So this enzyme is localized on the stroma side of membrane. Membrane. This is basically localized on the stromal side of the membrane, and along with the electrons that actually comes, from the acceptor of the electrons of the PS1, protons are actually necessary for reducing this NADP plus, that means NADP to NADPH. That means main reducing agents. All right. So you can say that along with electrons, the protons, that means this gradient that has been developed over here, this is very, very important for reduction of NADP. So along with electrons, the protons are very important for reduction of NADP to NADPH. So this is required for this purpose. All right. So we can say that. So let us take a look into the options, that what are the options that has been given. So this basically involves the breakdown of proton gradient. So definitely this is required. Proton gradient is definitely required for this one, ATP synthesis. Then there is breakdown of electron gradient. As such, we haven't heard or seen the electron gradient which is being developed. So we cannot say actually like that. So this is actually the incorrect statement. All right. So, this is actually the correct statement regarding the not true statement, if we are considering not true, because here we have to look out for the false statement. That's why. So, breakdown of the electron gradient is actually not true. This is false. And movement of the protons across the membrane to the stroma, again, this is also the correct statement. So, this is a true statement basically. All right, so this is also true. Then this is also true. However, this is false, which is a not true statement, we can say. And reduction of NADP to NADPH2 on the stromal side of the membrane, again, this we have also seen is a true statement over here, because for reducing it is actually being required. Because once the NADP will be reduced to NADPH, the electrons won't be generated. And those electrons, if won't be generated, then they won't be reducing, then they won't be creating a free, energy potential or a free reduction potential. And that free reduction potential is only responsible for pumping of the electrons from one carrier to another carrier. So that is the case. With this question, we can see that the statement B is the false statement. That means the not true statement. So with regarding to the question, this statement is the correct answer. That means option B is actually the correct answer that you can see over here. All right. So technically, we can say that the process doesn't involve the breakage of the proton gradient. Electron gradient. All right. Okay. Now let's move on to the next question, which is question number 12. Now, question number 12 says that habitat loss and fragmentation, over exploitation, and alien species invasion, all right, or extinction, they are the causes of. Now, we have heard these terminologies, you know, in the chapter, in the chapter of your biodiversity conservation, biodiversity and its conservation. And let me tell you that all these terminologies, that means habitat loss and fragmentation, overexploitation, alien species invasion, and co-extinction, they are actually the causes of biodiversity loss. So this is more or less a kind of a direct question that has been asked. So there are basically four major causes. So four major causes of biodiversity loss. Biodiversity loss. They are habitat loss and fragmentation, the first one, in which the habitat loss and fragmentation is the major one. Fragmentation, then overexploitation, then there is alien species invasion, and the last is co-extinction. Co-extinction. All right. So these are all the causes of biodiversity losses. So basically, statement that means option C is the correct answer. While population explosion, competition, natality, if you consider natality, so natality is basically a attribute of population. So this is an attribute of population actually. This is an attribute of population. That means a characteristic feature of population. All right, so this has nothing to do with all these features. Then there is competition. Now competition is one of the example of the interspecific population interaction. So this is an example of, you know, or you can say interaction, or an example of population interaction, because there are, unlike, just like competition only, there are other kinds of interaction also that happens in case of a population, whether it be mutualism or predation or parasitism or commensalism, etc., all those things. And regarding population explosion, so population explosion is also, we can consider to be as an attribute of population. This is also a characteristic feature of population. All right. So this has, again, this also has nothing to do with the all these above mentioned terminology. So option C is the correct statement over here, or correct option over here. All right. Now, question number 13. Now, question number 13 says that what is the net gain of ATP when each molecule of glucose is converted into two molecules of pyruvic acid? So it's actually talking about the net ATPs over here. All right. Now, let me tell you that during the entire process of glycolysis, so basically, glycolysis, this is indicative of the glycolysis process. All right. So, during glycolysis, you can say glycolysis, where one molecule of glucose actually gets converted into two molecules of pyruvic acid. Two molecules of pyruvic acid. That means 6 carbon. This is a 6 carbon molecule. Glucose is a 6 carbon molecule. So this will be breaking down into 3 carbon molecule of pyruvic acid each. This pyruvic acid is a 3 carbon molecule. All right. Now, during this entire process of, you can say, this one, glycolysis, a total of a total of 4 ATPs are actually being produced in this entire process. All right. So a total of 4 ATPs are produced. All right. So if you just look into the overall criteria, so a total of basically 4 ATPs are produced when one glucose molecule gets converted into two molecules of pyruvic acid. But out of these 4 molecule, if we are going to see, then out of these 4, 2 actually gets consumed in the reaction, and only 2 are being produced through substrate level phosphorylation. All right. So out of 4, you can say 4 ATPs, 2 ATPs actually gets consumed in the reaction, are consumed in the reaction. So if you just look into the net gain of ATP, so the net gain of ATP will be 2 ATPs only. The net gain of ATP will be 4 minus 2 is equal to 2 ATP. And the overall product, you know, the overall net gain of glycolysis is 2 ATP and 2 NADH. All right, so this is the overall net gain. This is in case of the anaerobic glycolysis. But if there will be aerobic glycolysis, then definitely this NADH will be reoxidized into ATP only. All right, so that's why the correct answer here will be option number C. That means 2 ATP. Clear? Now, question number 14. Now, question number 14 says that the girdling experiment that was performed by plant physiologist to identify the plant tissue through which. Now, this experiment is actually very popular for measuring or for accounting the amount of food transportation. All right. And this is basically responsible. This is one of the most popular experiments that has been performed in case of the plant body and is most widely being studied. And let me tell you that girdling experiment basically is related with the translocation of the fluid only. Translocation of the food, through the phloem tissue, and that too it indicates that the transportation, it actually takes place, in one direction only, because the food will be prepared by the leaf or the stem, green stems part, and that food will be ultimately translocated to different regions of the plant body, particularly towards the root part. That means the food will be translocated from stem or shoot towards the root. All right. So we can say that girdling experiment is basically, this also indicates that if a girdle is formed in the stem area, all right, if we just remove that particular tissue, phloem tissue, from that stem area, so this results into formation of, or the accumulation of the food in the form of girdle or in the form of circle in the stem area. All right. And because of which the food won't be able to translocate towards the root part, and so the roots will ultimately die. Clear? So we can say that girdling experiment shows the importance of phloem tissue, the importance of phloem tissue in the translocation of food from stem or leaf to roots of plants. All right. And this also shows, this also indicates or shows, you can say, shows that transport of food takes, so this also shows that the transportation or the translocation of food taking place in the plant body is actually considered to be as if the plant body is translocating food, so that food is basically considered to be the direction of the food is basically considered to be as bidirectional. All right, because phloem usually is responsible for translocating the food in in a bidirectional way. All right. So this also shows that transport of food takes place via phloem bidirectionally. Bidirectionally. That means if the food will be synthesized in the stem or leaf, you can say, then the food will be translocated towards the root for survival. And if the food will be synthesized, there are some plants where the roots, they are the storage organ or they are the synthesizing organ of food. So if the food will be synthesized in the roots part, then it will move towards the upward side. All right. So that's why it is bidirectional. So we can say that if we look into the different options, then we can say that is it related to water is transported? No. What food is transported? Yes. For both water and food transportation? No. Osmosis is observed? No. So that means option B is the correct answer. All right. Now, question number 15. Now, question number 15 says that which of the following is not observed during the apoplastic pathways? So there are certain statements that has been given over here regarding the apoplastic pathway. Now let me tell you, there are basically two important pathways through which the water and its dissolved solutes are translocated in the entire plant body. One is the apoplastic pathway and the other is the symplastic pathway. So apoplastic pathway, let me tell you, two ways of transporting water in plants. There are two ways, as I told you. The first one is considered to be as the apoplastic pathway or apoplast pathway. And the other one is considered to be symplast pathway. So apoplastic pathway is that pathway through which the movement of water actually takes place through cell walls. All right. That means the water movement is completely from outside. That is through cell walls. So there is no kind of membrane involved in this transportation pathway. You can say. All right. So we can say that this pathway involves movement of water through cell walls. The movement will be taking place through cell walls, and that too without crossing any membrane. Without crossing any membrane. All right. So this basically indicates that if there will be no membrane involved, so definitely, and if the water has to move, you know how actually the process goes on. That if you just look into the cells are actually arranged like this. All right, like this. So this is basically your epidermis, we can say. This is epidermis. Then epidermis is followed by cortex. Then endodermis. Then there is pericycle. Pericycle. And pericycle ultimately leads to, you know, xylem. Like this. All right. So if I'm talking about cell wall, if I'm considering it to be the cell wall, so the cell wall is basically arranged on the outer side like this. Like this. All right. So the movement of water is going to take place over here. So this is the cell wall. All right. So the movement of water will be taking place from here, except in the endodermis where there is a presence of Casparian strips over here. You know? Here is basically the presence of Casparian. The cell wall of the endodermis is actually suberized. It has the presence of suberin and it has the presence of actually Casparian strip, and that Casparian strip is suberized. So here the movement gets deviated, and then again it goes like this. The movement of water will go like this. So this is basically the movement of water through cell wall, and that too through apoplast I'm talking about. This is apoplast. All right. However, if the movement of water will be taking place through by crossing all the cell membranes inside, that means through here, you know, it has to pass all the cell membranes like this. So this is actually considered to be as the symplastic pathway. So this is symplast pathway where the cells actually passes the membrane. So you can just imagine that if a cell has to cross only the cell wall, so there is no kind of hindrance involved. None of the hindrances are involved from the cell organelles or any other constituents of the cell cytoplasm. So definitely we can say that apoplastic pathway is a continuous pathway and it's a faster pathway actually. All right. So we can say that it is a faster and a continuous pathway. So it is a faster and continuous pathway. While on the other hand, if we just see on the symplastic pathway, it is rather a slower process. It is rather a slower process. So we can just see, and symplastic pathway. So this involves water movement, by crossing cell membrane, by crossing membrane via, and that too they will be crossing a membrane through a very important junctions which is present in between over here, and that is considered to be as plasmodesmatal connections. All right. So via plasmodesmata. Plasmodesmata. All right. So that is the case. Clear? Okay. So let us take a look into the different statements that has been given. So is not observed, you have to find out during the apoplastic pathway. So movement of water occurs through intercellular spaces and the walls of the cell. Definitely this is a correct statement because I have already written over here, it occurs through cell wall. The movement does not involve crossing of cell membrane. Definitely this is also a true statement basically. Just a second. Yeah. So this is true statement and this is also a true statement. All right. Then the movement is aided by cytoplasmic streaming. This is a false statement because here the cytoplasm involvement is not the involvement of the cytoplasmic streaming is not involved because the cell doesn't process the membrane actually. And if a cell is not crossing a membrane, then how come it will be in contact with the cytoplasmic streaming? So this is a false statement. Then apoplast is continuous and does not provide any barrier to water movement. Again, this is also a true statement. So not observed, if we have to find out, then definitely this is a correct statement regarding to the question that has been asked. All right. So that is the case. Now let's move on to the next question, which is question 16. Now, question 16 says that which one of the following plants does not show plasticity? All right. Now, which one of the following plants doesn't show plasticity? Now, let me tell you that what is plasticity. So, plasticity is one such phenomenon in which there is basically changes in the shape of the leaf structures or any of the plant body structures because of changes in the environmental conditions actually. That means you can say that different kind of life form structures are being observed in case of the plant regarding to plasticity. So you can say that plasticity is defined as plants following different following, or you can also say adapting. So plants following or adapting different kinds of structures. So different kinds of structures during their life phase, life phases due to varying environmental conditions or due to response to varying environmental conditions. All right, so this is the definition. And one of the best examples of plasticity that has been given and that you might have also been studied is the example of plasticity is basically the heterophily that is observed in case of the plant body. So heterophily. Heterophily. Now, what is heterophily? Heterophily is basically the changes in the leaf structures depending upon the kind of adaptations they are acquiring. That means, depending upon the in their juvenile stage or in their vegetative stage, they will be having a different shape of leaf, and in their mature stage, they will be having a different shape of leaf. All right. So you can say that a phenomenon of showing different leaf structures, leaf structures in juvenile phase and mature phase. That means in the juvenile phase, the shape is different, and in the mature phase, the shape is different. Or in other words, you can say it completely depends upon the habitat. Like for example, if there is a plant which is found in a terrestrial habitat, so the leaf shape will be different. And if there is a plant which is found in aquatic habitat, so the leaf shape will be completely different. Leaf structure will be completely different. So that is the case. So mature, a phenomenon of showing different leaf structures in juvenile phase and mature phase, or in other words, plants having terrestrial adaptation shows different leaf structure, leaf structure, and those with aquatic life or aquatic adaptation shows different leaf structures. Clear? So this is the case. So one of the best example of heterophily the example of heterophily that has been given is found in larkspur, buttercup, and cotton. That is given in your NCERT. All right. So this is in cotton, larkspur, and coriander. Larkspur is basically buttercup, and coriander. All right. Now let us take a look at which options have been given. So doesn't shows plasticity. So cotton shows plasticity. Then coriander also shows plasticity. Buttercup also shows plasticity. As I told you, that last larkspur is buttercup only. All right. So maize doesn't shows plasticity. So that's why this is the correct statement. Correct option. Option D. Now, question number 17. Question number 17 says that DNA polymorphism, it forms the basis of. All right. Now, let me tell you that polymorphism is basically a phenomena of showing different genetic sequences, of you can say. So, DNA polymorphism is very, very frequent, and it is very, very common in case of genetic mapping also, and in the DNA fingerprinting also, because DNA polymorphism, it creates a basis of analyzing different DNA samples. All right, so this basically provides the way of different DNA samples. So sampling of the different DNA actually. So that's why this is one of the most basic principle of genetic mapping also, and in the DNA fingerprinting also, you can say. So we can say that DNA polymorphism in the DNA sequence forms the basis of both gene mapping, which is primarily being carried out in the human genome. So gene mapping or genetic mapping of human genome, you can say, and DNA fingerprinting also. DNA fingerprinting. because these DNA fingerprinting, they are primarily used in case of any kind of criminal cases or something in order to find out. So with the help of these DNA polymorphism, the sequences are being morphed, and so it is being found out that which DNA sequences is matching with which, you know, genes. So that's why if you just look into the options, option C is the correct answer over here. Clear? Now, question number 18. Now, question number 18 says that XO type of sex determination can be found in. All right. Now, let me tell you that XO type of sex determination is found in case of grasshoppers. All right. This is actually found in case of grasshoppers. We will be also seeing the other categories also, Drosophila birds and monkeys, because in case of Drosophila, if we see now, then Drosophila has the same kind of uh, chromosome constitution, just like that it is found in case of you know humans only. So though so Drosophila basically have just like they have a combination of human chromosome only. So they have a combination of XX and XY type of chromosome, where this is indicative of female chromosome, and this is indicative of male chromosome. All right. While in case of birds, if we see like chicken, if you're going to see regarding that part in case of birds, so they have a combination of ZW and ZZ type. So they have ZW and Z type of chromosome, where the ZW, so basically the ZW here is indicative of female. That means they show female heterogametic. All right. Here there was male heterogamety, as you can see over here. The gametes are different. So this is indicative of male heterogamety, because the males are having two different sets of chromosomes. While here the female is indicative of this and the males are indicative of ZZ type. So that's why female heterogamety. And C option, which is of grasshopper option, if you're going to see. So grasshopper is having a combination of XO and XY type of chromosome, where this XO, that means, is indicative of female. So basically females, they lack one chromosome, one X chromosome. All right. So they have only one chromosome of X. All right. They have only one X chromosome besides the autosome. In case of right, so in case of grasshopper, if we are going to see, so basically the combination is of XO and XX type, XX type basically. So here it will be indicative of this factor. So the XO combination is basically being represented by, that means this XO combination O basically signifies that there is a lack of one there's a lack of one Y chromosome, or there's a lack of actually Y chromosome, you can say. All right. So this is indicative of males over here, and this is indicative of females over here. So that means males, they usually lack one chromosome. All right. So males have only one X chromosome besides the autosome, while the females will be having a pair of chromosomes. And the last is monkeys. So monkeys also, they have the same pattern. That means XX and XY type of chromosomes are being found. So female and then male. XY will be indicative of males. Clear? So out of all the options that we can see over here, option C is the correct statement. Correct answer. All right. Now, let's move on to next question, which is question number 19. Now, question number 19 says that you have to uh, identify the reason and assertion statements have been given, and you have to choose the correct statement from the options below, given below. Now let us take a look. All right. Assertion A and the other is labeled as reason R. Now assertion A says that polymerase chain reaction is used in DNA amplification. Okay. Now the reason that has been given for this statement, for this assertion, is that the ampicillin resistant gene is used as a selectable marker to check the transformation. All right. Now, in the light of the above statements, you have to choose the correct statement from the options given below. All right. Now, if I just look into both the statements, if you just take a look into the both statement, then the option A says that, which is PCR, that means polymerase chain reaction is used in DNA amplification. So this is definitely a correct statement. All right. That means A is a correct statement. All right. Because A is actually a correct statement, because PCR is one such mechanism that increases the copy number of DNA via DNA amplification. So PCR is a chain reaction that increases the copy number of DNA, yeah, of a target DNA, you can say, rather, of a target DNA via DNA amplification process. So this is a correct statement. Clear? Now let us take a look into the second statement. Now, second statement says that ampicillin resistant gene is used as a selectable marker for the transformation process. Now, again, this statement is also considered to be as a correct statement, because ampicillin resistant gene, ampicillin is basically an antibiotic. All right. And this antibiotic resistance gene can be considered, or it is actually considered or used as a selectable marker in order to differentiate between the transformed cell and the non-transformed cell. All right. So this statement is also correct. That means R is also correct. R is also correct. That means antibiotic resistance gene resistant gene is used as selectable marker. So this gene is actually used as selectable marker for differentiating between transformed cell from the non-transformed cell. For differentiating transformed cell from non-transformed cells via the process of transformation. All right. So via transformation. All right. So both statements are correct. But let me tell you that these two statements that has been given, both the statements are considered to be correct. But this R, that means the reason that has been given for the assertion, is actually not correct. That is not the correct explanation of A, because this assertion is a completely different process. That means PCR is a completely different process where the DNA copy number is being increased via amplification process. While the transformation process is a completely different thing where cloning vectors are being used. All right, where in this transformation process, it's a completely different process. It has nothing to do with the PCR mechanism or the DNA amplification process. So it's a completely different process that in which the cloning vectors are being used and selectable markers are being used for creating a transformed and a non-transformed cell. So that's why we can say that both A and R are correct, but R is not the correct explanation of A because both these two are different methods. So you can say, however, both these methods are completely two different methods. Two different methods having no correlation with one another. Correlation with one another. So that's why the correct answer will be option number B over here. Statement. All right. Now, let's move on to question 20. Now, question number 20 says that the gaseous plant growth regulator is used in the plants too. Now, let me tell you that the gaseous plant regulator, which is found in the plant body, plant growth regulator actually, plant growth regulator in the plant body is ethylene. Is ethylene, which is denoted as C2H2. All right. And ethylene is considered to be as a gaseous plant hormone, and it has some very important aspects. All right. And one of the most important aspects that they are primarily being required, in for the ripening process, or for speeding up the ripening process. Then they also promotes female flowering in the pineapple plants. So there are a lot of options and a lot of functions of this ethylene hormone. So let us take a look that what are the options first that has been given, and we can just sort out the options. So speeding up the malting process. Now this is one such characteristic feature. This feature basically belongs to gibberellin hormone. This is not a feature of let me tell you, this is not the feature of gaseous PGR, that means ethylene, but this is a feature of gibberellin hormone that speeds up the malting process in the brewing industries. All right. Then it promotes root growth and root hair formation to increase the absorption surface. All right. Okay. So this statement has been given. We will be coming back to the statement. It helps in overcoming the apical dominance. Now, this is one such feature that belongs to the, or that this is one such function that belongs to the plant growth regulator cytokinin. So cytokinin is one such hormone that helps in overcoming the apical dominance, which is promoted by auxin. So definitely this is not a correct statement. All right. Now this kills the dicot weeds in the fields. Now this is a function of auxin. This is one of the important functions of auxin, and that too synthetic auxin, you can say, because two categories of auxins are found: natural and synthetic. So this is one of the features of auxin. So the only option left with us is basically this one, that means option B over here. Now let me tell you that why it promotes the root growth and root hair formation to increase the absorption surface, because sometimes ethylene is also responsible for being secreted in the plant body during adverse condition or any kind of harsh condition, you can say. All right. So that's why it results into adventitious roots formation in the plant body. So ethylene, it basically promotes, this promotes root growth and root hair formation So root growth and root hair formation for increasing the absorption surface during water deficient conditions. Water deficient condition. That means when there is less availability of water. So this basically is responsible for inducing the root growth also and root hair formation also, so that the absorption area and the surface area of the root growth, root hairs and root grow, main root gets increased, and that's how they will be able to absorb more and more amount of water during the water deficient condition. All right. So that is the case. So that means option B is the correct answer. All right. Now question number 21. Now question number 21 says that the device which can remove particulate matter that is present in the exhaust from a thermal power plant. All right. Now the device that is responsible for removing the particulate matter. Particulate matter, basically the suspended particulate matter, you can say, that is found in the exhaust from a thermal power plant. Now, out of all the options that has been given over here, all right, so this one, that means every option, that means STP or incinerator or electrostatic precipitator or catalytic converter. Basically, STP, let me tell you regarding the STP first. So STP here stands for sewage treatment plants. All right. So this basically stands for sewage treatment plants. And this has nothing to do with the removal of particulate matters over here. All right. Because since this is a sewage treatment plant, so it will be treating only the sewage. And here it is asking about the exhaust from the thermal power plant. So definitely this is not the correct statement. This is not the correct option, you can say. All right. Then incinerators are basically a kind of a burner system, you can say, and these burner systems, they are this basically involves burning the garbage or burning huge dumps of waste at a very high flame or at a very high temperature. So this is basically a burning of garbage at a very high temperature. So burning garbage at high temperature. So again, this has nothing to do with removal of particulate matter from the exhaust, because here the temperature factor plays a very important role. So burning wastes at high temperature, or you can say very high temperature, in fact. Very high temperature. Clear? Then the third is the electrostatic precipitator, and the fourth is the catalytic converter. Now, let me tell you first about the catalytic converter. So, catalytic converter again has a very, very different role. So, this is also having an again very different role, because catalytic converters are the devices which are actually fitted in the automobiles for removing the exhaust from the vehicles. All right. Or for removing the nitrogen containing oxides from the vehicles, but not from the thermal power plants. All right. So you can say that catalytic converters, these are fitted in the automobiles. So these are fitted in automobiles. That means vehicles. For reducing the emission of poisonous gases. All right. So that is the case. So they basically convert the poisonous gases into harmless gases, harmful gases into harmless gases once the fumes passes through this converter, and that's why. So this is again also not a very good option. So definitely this is not the case. And so the only option left over here is the electrostatic precipitator. Now let me tell you that electrostatic precipitators are those devices that is actually responsible for removing approximately 99% of the particulate matter that is found in the exhaust of the thermal power plant. So they uses a technique or a method of flow of electrons. All right, there is a setup of electrodes and there is a flow of electrons through this entire setup. And those electrons, they actually trap all those, they provide negative charges to the dust particles, and they trap all those particulate matters into those electrodes. And then there is a shielding plate over there that holds it. So it's a completely entire different setup. So electrostatic precipitator helps in removing 99% of particulate matter from the exhaust of thermal power plants. All right, so this is the case. So option C is the correct answer. Now, question number 22 over here. Now, question number 22 says that again, given below are the two statements that has been given, and you have to, in the light of the above statements, you have to choose the correct answer from the options given below. Now you have to select the correct statement from the options given below. All right. Now statement 1 says that cleistogamous flowers, they are considered to be invariably autogamous. All right. And the statement 2 says that cleistogamy is disadvantageous as there is no chance of cross-pollination. Now let me tell you that basically cleistogamy is one such process, or cleistogamous flowers are basically those flowers. It is one of the categories of autogamy. All right. So in case of autogamy, if you see, there are two types of flower that are generally being found in autogamy. One is the chasmogamous flower and the other is the cleistogamous flowers. Chasmogamous flowers, and then there is cleistogamous flowers. All right. Now, chasmogamous flowers are those flowers so which have well exposed anthers and stigma. So since they have well exposed anthers and stigma, so that's why it is open to both self pollination also and cross pollination also. So so they have well exposed anthers and stigma. Anthers and stigma. While on the other hand, the cleistogamous flowers are those flowers which actually do not open at all. They are closed flowers. So they are completely closed, and so they do not open at all. So these flowers do not open at all. Open at all. And imagine if these flowers won't be opening at all, then that's why it is considered to be as invariably autogamy. Invariably autogamy means they will be invariably only and only self-pollinated. They won't be having any chance or any by chance of undergoing the process or having the process of cross-pollen landing. So that's why they are invariably autogamy. They are invariably autogamous. Invariably autogamous. That means they can only be self-pollinated. self-pollinated. Yeah, so they can only be self-pollinated, and since they can only be self-pollinated and they do not open at all, so that's why there is no chance of cross-pollen landing also. So there is no chance of cross-pollen landing. So that means no chance of cross-pollination. And if there will be no chance of cross-pollination, so definitely this is one of the disadvantageous factor for this flower, because at one point or after few generations, because of repetitive self-pollination, it will become sterile. All right, self-sterile. So that's why there's no, since there is no chance of cross-pollen landing, so this is a disadvantageous factor. Disadvantageous factor for this flower. All right. Now let us take a look into the statements. So with this, we come to know that actually both the statements are considered to be as the correct statement. All right. So if we just look into the options, so option option A, which says that both statement, that means statement 1 and statement 2, they are correct. So that means option A is the correct answer over here. All right. Now let's move on to question number 23. Now question number 23 says that the exoskeleton of arthropods is composed of. All right. Now exoskeleton of arthropods, let me tell you that basically arthropods are the categories of insects. All right. So this is the largest phylum of the animal kingdom, arthropods. Arthropods are the category, just a second, category of insects. All right. However, marine arthropods are also being included over here, but mostly they are insects only. And since this is talking here about the exoskeleton of arthropods, so the exoskeleton of arthropod is basically, we have studied it also in the animal kingdom chapter, that the exoskeleton of arthropods is chitinous. That means it is made of chitin. So exoskeleton is chitinous. That is made of chitin. Made of chitin. All right. And chitin is basically a composition of N-acetyl glucosamine. Let me tell you. Sorry, this chitin is basically, glucosamine is a different thing, but this is a polymerization of a complex polysaccharide, let me tell you. So that's why we can say that this exoskeleton of arthropods, which is generally made of this chitin, and this chitin is also one of the important constituent of the cell wall of fungi. All right, so an important constituent or an important constituent of fungal cell wall, you can say. All right, so that is the case. So option C is the correct answer over here. While cutin, if you are going to see, so cutin is basically a different thing. It's a kind of a cuticle-like structure which is found on the epidermal surface of the this one. It's a basically a kind of a, you can say, a derived lipid, or you can say considered to be as a complex lipid molecule. So again, this is not a correct option. Cellulose is basically found as a cell wall component of plant cells. All right. So this is a polysaccharide. So again, this is not the correct statement, because this is found in case of the plant cells. And glucosamine is basically a monomeric unit. All right, so this is also not an option because, and this is a composition of bacterial cell wall because bacterial cell wall is made of peptidoglycan, and peptidoglycan is a combination of NAM and NAC. That means N-acetylmuramic acid and N-acetylglucosamine. So that's why glucosamine also is not the option. So the correct answer will be option C over here. All right. Now let's talk about question number 24. Now, question number 24 says that you have to read the following statement and you have to choose the set of the correct statements over here. All right. Now, let us take a look at which statements are correct and which statements are actually incorrect. Okay. So, the option A says that euchromatin is loosely packed chromatin. All right. So basically, there are two types of chromatin. You people might have heard it also. One is the euchromatin, and the other is the heterochromatin. So euchromatin is basically here. If I say, just a second, then euchromatin is a loosely packed chromatin. This is true. Is a loosely packed chromatin, and it is considered to be as transcriptionally active. Loosely packed chromatin, which is lightly stained also. Lightly stained. It is having very light staining. So lightly stained. And anything which is loose, so definitely it will be transcriptionally very active. Why? Because it will provide the spaces for the insertion of those essential enzymes, proteins, and all those things that will help in the process of transcription, that will carry out the process of transcription with very ease. So that's why, since they are loosely packed, so that's why they are transcriptionally very active. So they are transcriptionally active. Clear? Like this. But the other category of chromatin, which is considered to be as heterochromatin, heterochromatin, on the contrary to euchromatin, is densely packed. So heterochromatin is actually densely packed chromatin, and anything which is densely packed, they definitely it will be having a darker stain. So they are darkly stained, and most importantly, they are transcriptionally inactive because they won't be providing spaces for the accommodation of those essential proteins or enzymes or important essential genes. So the genes will be completely inactive over here in case of the heterochromatin. It's actually chromatin. Yeah. So transcriptionally inactive over here. Clear? So we have seen the statements now. So with this, we come to know that this is the correct statement. All right, this is the correct statement. And heterochromatin is transcriptionally active? No. Heterochromatin is basically the incorrect state. This is the incorrect statement because heterochromatin is transcriptionally inactive. It is not active because of dense packing. Now, histone octamer is wrapped by a negatively charged DNA in the nucleosome. Again, if we just look into the nucleosome structure, so nucleosome is basically defined as a structure in which negatively charged DNA, negatively charged DNA. Now, why negatively charged DNA? Because DNA is having phosphate group at its 5' end. So that's why it is negative charge. So negatively charged DNA is wrapped around positively charged histone octamer. So positively charged histone octamer. All right. So this is a nucleosome structure. So this is a true statement. So this is also a correct statement. All right. Now histones are rich in lysine and arginine. Again, this is also a correct statement because histone, which are actually the basic protein. This histone of tamer is a basic protein. This is basically a basic protein, and definitely it is rich in two amino acids, which is lysine and arginine. So this is rich in lysine and arginine, two basic amino acids. Arginine. These are basic amino acids. So again, this is also a correct statement. That means this one. Now, a typical nucleus basically contains, a typical nucleosome basically contains 400 base pairs of DNA helix. It is talking about. That means if you just look into the nucleosome structure basically contains not 400, but typically you can say that it contains 200 base pairs of DNA helix. So this is not the correct statement. This is actually the incorrect statement over here, because nucleosome contains nucleosome contains 200 base pairs of DNA helix. Clear? So this is the incorrect statement. So A, then C, and then D is considered to be the correct statement. So if you just look into the options, option B over here is considered to be the correct answer. All right. Now, the next question is that which one of the following statements cannot be connected to predation? All right. Now, predation is one of the category of population interaction actually. All right. So let me tell you about predation first. So predation is a type of population interaction. All right. And it is one such interaction where one organism is benefited and the other organism is harmed. All right. So the organism which is benefited is the predator. All right. And the organism which is harmed, that loses the life, is basically the prey species. So a type of population interaction in which one organism is benefited which is predator. All right. While the other is harmed. Harmed, which is prey species. All right. So this is the case. Now, predation, if you just look into the options that has been given, so it helps in maintaining the species diversity in a community. Definitely, we can say that predator basically helps in maintaining species diversity in the community, because a predator never only feeds on only one prey. So it maintains the species diversity in a community by never feeding only on a single prey, or by never attacking on any particular category of prey. So that's why they help in maintaining the diversity in a community. So you can say that predation or predator helps in maintaining species diversity. So how it will be maintaining species diversity? By actually reducing the competition among the other prey species. So by reducing the intensity of competition because they never feed on only single prey species among other prey species. So they definitely maintain the uh, species diversity in a community. Then it might lead to extinction of a species. No, if the diversity is maintained in a community, then definitely it doesn't lead to extinction of a species. All right, why? Because if a predator, if the predator is so much efficient, all right, that it is never going to exploit its prey, and that's how it never leads to extinction of a particular species. All right, so predator is too efficient that it never exploits or never over exploits, you can say. Over exploits its prey. And so the species cannot become extinct. So species actually cannot become extinct. All right. So this is actually, you can say, it might lead to extinction of a species it is talking about. Now, so again, this is also a correct statement. Because if it is too efficient, that it is never going to over exploit its prey, so the species can't become extinct. But if a predator, you can say that if a predator is too efficient, or you can say that that predator can kill up all the species, then definitely the prey might become extinct also. So we can also write that if a predator over exploits its prey, over exploits its prey, then it might become extinct. Might become extinct. All right, so this is the case. Now, both the interacting species are negatively impacted? No. As I already told you that this is an interaction where one species is benefited and the other is neither benefited, and the other is harmed. So definitely, this cannot be connected to the predation. So this is a wrong statement. Now, it is necessitated by the nature to maintain the ecological balance. Now, definitely, predation, since this is one such category of population interaction, so that's why it is very, very much necessary in the nature. This interaction is very, very much necessary in the nature to maintain the ecological balance. All right, between the prey and the predator species, because these organisms are a part of our food web, and if these organisms will be eliminated, then definitely this will disturb the entire ecological balance. All right, so definitely they are very, very important in our nature to maintain the ecological balance. So with this, we can say that statement C cannot be connected to predation. So the correct answer is this one only. This is the correct answer. Clear? That means option C. So let's talk about next question, which is question number 26. So question number 26 says that the flowers are zygomorphic in. All right. So basically it is asking about the symmetrical pattern of the flower combination. All right. So in this category, now let me tell you that what is zygomorphic. So zygomorphic is basically a category of symmetry. All right. So zygomorphic symmetry, which is also considered to be as bilateral symmetry. So this is relevant, or this is synonym to bilateral symmetry. In case of the, in case of animals, we consider it to be as bilateral symmetry. Now, zygomorphic symmetry is, or which is also known as bilateral symmetry. This is one such symmetrical pattern in which a flower gets divided into equal halves or similar halves when it passes through a central plane, or when a flower is being crossed through a central or a vertical plane. All right. So it is defined as, when a flower gets divided into equal halves. So when a flower gets divided into equal halves, just a second, passing through a central or vertical plane, you can say. Vertical. All right. So that is the case, and this the opposite of the zygomorphic symmetry is the actinomorphic flowers. That means, in case of the actinomorphic flowers, the flower, it can be divided, or it basically gets divided into equal halves, passing through any of the central plane when you will be dividing it. All right. So if any of the plane of axis can be given out, so that will result into the division of the flower into equal halves. And this is in case of actinomorphic I'm talking about. All right. So the best example of this zygomorphic pattern of, symmetry is prominently being shown by, plants like pea. Then there is gulmohar. Then there is cassia. All right. And beans. These are the prominent examples of zygomorphic symmetry. All right. Now let us take a look that what are the options that has been given. So this is mustard, gulmohar, cassia, datura, and chili. So out of all these options, that means the matching up options basically belongs to only two categories, and that is gulmohar and cassia. That means this one, which is B and C only. So our correct answer will be B and C only. While rest of the three categories, that is mustard, datura, and chili, they belong to actinomorphic flower. While mustard, datura, and chili, they are actinomorphic flowers. It's actinomorphic flowers. That means these flowers can be divided into equal halves, passing through any plane of axis. All right. Now, so answer B is the correct answer. Now let's move on to question number 27. Now question number 27 says that which one of the following produces nitrogen fixing nodules on the roots of alnus? All right. So it's talking about the root nitrogen fixing nodules on the alnus, roots of alnus. Now let me tell you first about alnus. That alnus is an angios. It's a category of basically your non-leguminous plant. All right. This is an angiospermic family belonging to the monocot, but that too in monocot we might have heard it about it. These nitrogen fixing nodules that primarily fixes nodules in case of the leguminous plants. But alnus is a non-leguminous plants. This is an example of non-leguminous plant. Just a moment. So this is an example of non-leguminous plant. And in case of this alnus, the one category which is responsible for fixing the nitrogen is a free living actually bacteria, and that is called as Frankia. All right. So the microbe Frankia is primarily responsible for fixing the nitrogen in the root nodules of the non-leguminous plants. So Frankia fixes nitrogen in the root nodules of non-leguminous plants. While on the other hand, if you see the other different categories, that means Rhizobium, Rhodospirillum, and Biseringia. So Rhizobium is basically a nitrogen fixing symbiote in the leguminous plants. All right. So Rhizobium is a nitrogen fixing symbiote. Nitrogen fixing symbiote in leguminous plants. So this is a nitrogen fixing symbionts in all the leguminous plants. All right. And on the other hand, this Rhodospirillum and Biseringia, these are free living nitrogen fixing bacteria. So Rhodospirillum and Biseringia. Biseringia. These are free living nitrogen fixing microbes or bacteria. Nitrogen fixing bacteria. All right. So that is the case. So that's what the correct answer is, Frankia oil, option B. Now question number 28. Question number 28 says that you have to read the following statements about the vascular bundles. And so they basically 5 statements have been given, as you can see over here. And you have to choose the correct answer from the options that are given below. All right. Now let us take a look one by one, at the different questions or at the different statements that has been given, and we'll just sort it out that which statement is correct and which statement is incorrect. So in the roots, the xylem and phloem in a vascular bundle, they are arranged in alternate manner along the different radii. So this is in case of roots, if we see, all right. So in case of roots, the vascular bundle which is found is basically it is having the arrangement like this. Then again this one, then this one. All right. So this is something, the figure turns out to be like this. All right. And this is the xylem part that we are drawing with. So this is basically the xylem part. This is the xylem, and this is the phloem. So you can see that the xylem and phloem, they are actually arranged in alternate manner. All right. So xylem and phloem arranged in alternate manner, and that's why this is considered to be as radial vascular bundle. So radial vascular bundle is basically an arrangement in which the xylem and the phloem, they are arranged alternatively to one another. All right. So this is radial vascular bundle over here. So this is a true statement actually, because they are arranged in alternate manner. Now the next statement says that conjoint closed vascular bundles, they do not possess cambium. This is again also true that basically there are two types of, let me tell you that there are two types of vascular bundles. All right. So two types of vascular bundles are found. Vascular bundles are there. One is considered to be as the open vascular bundle, and the other is considered to be as closed vascular bundle. All right. So open vascular bundle is one such vascular bundle which will be having the presence of cambium in between the xylem and phloem like this. All right. So here above will be the phloem part. This is the xylem part that I am drawing, and this layer is cambium. So it is having the layer of cambium in it. But in case of closed vascular bundle, if we see, we do not have the presence of, or the vascular bundle, they do not have the presence of any kind of cambium, and therefore no secondary growth is also seen. But since in case of open, the cambium is present, so secondary growth is also found. All right. And this is arranged like this. So you can see there is no cambium. So this is the phloem. This is the xylem. Clear? Here, no cambium is found between, no cambium is found between the closed vascular bundle, in between the xylem and phloem. So definitely this statement is also correct. All right. B is also correct. Then in case of open vascular bundles, cambium is present between the xylem and phloem. Again, this is a true statement. We have seen that in case of this open vascular bundle, the cambium is being found. So C statement is also correct over here, as we have seen over here. All right. Now D statement says that the vascular bundles of the dicot stem possesses endarched protoxylem. All right. Now dicot stem, if you're going to look about the dicot stem, so in case of the vascular bundles of dicot stem, they have an endarched protoxylem. Now let me tell you that what is endarched protoxylem. So in case of the endarched protoxylem, the xylem is arranged towards the pith, and the phloem is arranged towards the periphery. All right. So, and that too protoxylem. So endarched basically means xylem arranged towards pith, and that too, this xylem is also of two categories. One is called as the protoxylem, and the other is called as metaxylem. All right. So here the protoxylem is arranged towards pith, I'm talking about in case of endarched arrangement. All right. So while in case of the exarch arrangement, the protoxylem is towards the pith and the metaxylem is towards the exarch. Just a second. Exarch. So in case of exarch arrangement, metaxylem towards pith. And this endarch arrangement is found in stem. An exarch arrangement is found in roots. So metaxylem towards pith and protoxylem towards periphery. Periphery. All right. So this is the case. So with this statement, we come to know that this statement is also correct. All right. Then the last statement is in monocot roots. Usually more than 6 xylem bundles are present. And this is also a correct statement because in case of monocot root, you know, in monocot root, polyarch condition is being found. So in monocot roots, since polyarch condition is found, that means vascular bundles are more than, vascular bundles are actually more than uh, 4 or 5. So that's why polyarch condition. So since polyarch vascular bundle is being found, vascular bundle is found. So you can say that 6 xylem bundles are more. So more amount, or rather we can say more amount is basically the 6 number. Six numbers of xylem bundle. Xylem found. Present. So this is also a correct statement indirectly. All right, this is also correct. So that means altogether all the statements are correct, A to E. So let us take a look. So A, B and D only, B, C and D only, A, B, C and D only, and A, C, D and E only, and then none of the above. So definitely, since all the statements are correct, so it is not matching with any of the above options. So there's only one option left, that is none of the above. All right, because all the statements are correct, and that's why this A, B, C, D, E combinations has not been given. So that's why the correct answer is none of the above. All right, now question number 29. Now question number 29 says that you have to identify the incorrect statement regarding to the pollination. All right, so pollination, we all know that it's a process of transfer of pollen grains from anther to stigma of a flower that can be mediated by the biotic agent also or by the abiotic agent also. Now let us take a look into the different options. Pollination by water is quite rare in the flowering plant. So again, this is a correct statement that the pollination by water is rare in angiospermic plants, or you can say flowering plants, because it is only limited to around 30 genera of monocotyledons. That's why. As it is limited to 30 genera of monocots. However, in case of the dicots, if you will see, so they are more prominently inclined towards, biotic pollination. That means insect pollination rather than the water pollination. And water pollination is more common in case of your lower plants like Algae, Bryophyta and Pteridophyta. So there it is very, very common. But however, it is quite very rare in flowering plants. All right. Now, pollination by wind is more common amongst abiotic pollination. All right. So definitely, there are two kinds of abiotic pollination that we have seen. So two types of abiotic pollination. Abiotic pollination. All right. So the one is being carried out by wind, which is called as anemophily, and the other is being carried out by water, which is called as hydrophily. However, this wind one is considered to be as more common than water pollination, because water is water is more applicable in case of lower plants, but not on the categories of higher plants. All right, so this is the correct statement again. Now, flowers produces foul odors to attract flies and beetles to get pollinated. All right. So again, this is also a correct statement because we have heard it also, and we have you might have studied it also, that flowers that are being pollinated by flies and beetles, they secrete foul odor. So flowers pollinated by flies and beetles secretes foul odor. Foul odor. All right. So this is also true. That means this is true, and this is true. So the only option, incorrect option that is left is option D over here. So option D is saying that moths and butterflies are the most dominant, pollinating agent among insects. Now, this is an incorrect statement because this statement, in this statement, it is asking about the most dominant pollinating agent. And the most dominant pollinating agent among insects are the bees. Bees are the most dominant pollinating agents. So dominant biotic pollinating agents. So that's why option D will be the incorrect statement with regard to question number 29. So that means the correct answer will be this one. All right, option D over here. Now, question number 30. Now, question number 30 says which one of the following statement, all right, is not true regarding the gel electrophoresis technique. All right. Now, gel electrophoresis technique, let me tell you that gel electrophoresis is one such technique of biotechnology that helps in, separating the DNA fragments, all right, on the basis of their molecular weight by passing electric field through them. So, if we just go through the different options, all right, so we can just first write that gel electrophoresis is a method of separating DNA fragments on the basis of their molecular weight. Molecular weight when passing through an electric current. Passed through electric field. All right, so that is the case of, or that is the definition of gel electrophoresis. And here agarose gel is being used for sampling the DNA. All right. So the first statement says that the process of extraction of separated DNA fragment from the gel is called as elution. Now, this is a true statement. This is a very true statement that the process, once after the DNA gets separated, the separated DNA is extracted through the process of elution. So, once after DNA is separated, DNA is separated. Once after DNA is separated, they are actually stained. So then comes the staining procedure. Staining of DNA is done. Staining of DNA done. And then it is being visualized under UV rays. Visualized under UV rays. And this will give an appearance of orange bands. So appearance of DNA as orange bands. And after this, once they are being appeared in the form of orange band, then after this, through the process of elution, they will be extracted. DNA extraction via elution. Clear? So this is how the process is being carried out. So let us take a look into the statements. So the separated DNA fragments, they are stained by using ethidium bromide. Yes, this is also a correct statement because the staining here is being done with the help of a stain that is called as ethidium bromide, which is shortly termed as ETBR. Ethidium bromide. So the staining is done with this stain only. That means ethidium bromide. So this is a correct statement. Now the presence of chromogenic substrate means the DNA only, gives blue colored DNA bands on the gel. Now this is incorrect because we have seen over here that the DNA will be appeared in the form of orange bands, not in the form of blue bands. So that's why this is an incorrect statement. And bright orange colored DNA bands can be observed in the gel when exposed to UV light. Now this is a true statement. All right. So out of all the statement, which is the false statement, so this is considered to be as the false statement over here. All right. So that's why option C is the correct answer. All right. Now, question number 31. Now, question number 31 says that the production of cucumber has increased manifold in the recent years. Now, application of which of the following phytohormones has resulted in this increased yield, as the hormone is known to produce female flowering in the plants? Now, this is a very, very direct question that has been asked from your plant hormone section, because there are some applications that are required to be known. All those applications of different plant hormones has already been mentioned in your NCRT. So this is a very, very direct question because here the keyword has been given that in case of cucumber, female flowering is being induced. So in cucumber, female flowering is induced. Female flowering is induced. Now, this female flowering, it can only and only be induced with the help of a hormone, and that is called as ethylene. Because this is the only hormone that induces female flowering, and that too in cucumber via ethylene hormone. Ethylene hormone. So the most commercial available form of ethylene is basically the ethy form that is being commercialized. All right, so that is the case. Since it is talking here about the female flower. Clear? While ABA, gibberellin, and cytokinin, they also induces flowering in the plant body, but they do not induce a specific kind of sexuality in the plant body. They will promote flowering. All these promotes flowering, but they do not provide any kind of particular sex kind of a flowering in the plant body. All right. So that's why option C is the correct answer. Now, question number 32. Now, question number 32 says that what amount of energy is released from glucose during the lactic acid fermentation? All right. So let me tell you that during the process of the lactic acid fermentation, now lactic acid fermentation is basically the category of anaerobic fermentation, let me tell you. And this fermentation occurs in case of the mussels. All right. So lactic acid fermentation, and this is one such fermentation that belongs to anaerobic respiration, is a category of anaerobic respiration. That means there will be no involvement of any oxygen over here, and therefore the carbohydrates, in case of the lactic acid fermentation, the carbohydrate gets once the glycolysis process is being carried out, that glucose will be converting into pyruvic acid via glycolysis, and that pyruvic acid, depending upon its availability, will be converting it into the lactic acid. All right. So lactic acid fermentation occurs in mussels. This occurs in mussels and lactic acid bacteria, which is lactobacillus. which is lactobacillus. All right. And so this is again a direct question that has been asked. And let me tell you that since lactic acid fermentation is an anaerobic respiration, it's a category of anaerobic respiration. So you people might have been knowing it also, that in case of anaerobic respiration, very less amount of energy is being released. All right. And that energy is only available for utilizing, for being utilized by the particular bacteria or by the particular cell, not for producing further energy. All right. So this is again a very direct question, and it's a kind of an analytical question that has been asked. All right. So if you can just use a common sense, this can be sorted out. So that's why I have written here anaerobic respiration, where the energy released is already very less. Energy released here, released is very less, and this is approximately around 2 ATPs of molecules are being released. All right. So we can say that it is equivalent to, we can say it is equivalent to 21 kilocalorie or 2 ATP, you can say equivalent. Clear? So that's why out of all the options that you can see over here, this one, approximately 50%, this will be a very great energy. So this won't be an option. More than 80%, again, this won't be an option. And more, about 10%, again, this won't be an option. So less than 7% of energy is being released. So less than 7% of energy in glucose is released during lactic acid fermentation. All right. So option D is the correct answer. Now, question number 33. Now, in case of the question number 33, there are certain statements that have been given below, and you have to choose the correct answer from the options that has been given. All right. Now, let us take a look. That option A says, in the old trees, the question says that in the old trees, the greater part of the secondary xylem is dark brown and resistant to insect attack. Definitely, this is a true statement, because in the old trees, the greater part of the secondary xylem is actually darker brown and it is resistant to attack. So, what is the actually reason why it is being there? So, it is due to secretion of secondary metabolites and their deposition in the lumen of the vessels. All right. Or deposition of organic compounds like tannin and resins in the central layers of stem, then deposition of suberin and aromatic substances in the outer layer of stem, then deposition of tannin, gums, resins, and aromatic substances in the peripheral layers of stem, and presence of parenchyma cells, functionally active xylem elements, and essential oils over here. All right. Now let me tell you that this category, that means in case of the, if it is here talking about the this one, that means the greater part of the secondary xylem. Now secondary xylem, which is also considered to be as the bark of the tree. All right. So this is mostly you can say mostly dark brown. It is because of the presence of the deposition of organic compounds. All right. And all those organic compounds, they are usually localized. Organic compounds means what? Organic compounds means tannins, resin, lignin, oils, gums, all those cases. All right. So the secondary xylem, you can say in old trees, secondary xylem turns dark brown. dark brown due to deposition of, so it is due to deposition of organic compounds, like, so it is because of the deposition of organic compounds like tannins. And let me tell you that all these substances, tannin, resin, gum, oils, these are all secondary metabolites only. So tannin, resins, gums, oils means essential oils. All right, aromatic substances, all those things. Aromatic substances in the inner layers of stem. Inner layer means the central layer. So in the inner layers, or you can say central layers. Central layers of stem. All right. And as I already told you that all these above substances are secondary metabolites. That means they will be only synthesized in the plant body whenever they are required, not through the entire lifetime. All right. So that's why, and it is already deposited, since this is the deposition is being done in the secondary xylem. So that too in the secondary xylem, they are deposited in the cavity of the vessel. So all these substances, their deposition is being done. Secondary metabolites are deposited in the cavity of secondary xylem, the cavity of the vessels of secondary xylem. In the cavity of vessels of secondary xylem. All right, and that's why it makes it very, very hard and resistant for durable also for other microorganisms and other adverse conditions to deter it. All right, so let us take a look again now. So secretion of secondary metabolites and the deposition in the lumen of the vessel. Now this is true. Then deposition of organic compounds like tannins and resins in the central layers of stem. This is also true. Now deposition of suberin and aromatic substances in the outer layer. Now this is false because suberin is a water impermeable substance. All right. So this has nothing to do with the resistant part. So suberin won't be there because it is a water resistant substance that is found in endodermis. All right. Then deposition of tannin, gum, resin, and aromatic substances in the peripheral layers of stem. Now, as it is saying, it is asking about the peripheral layers, but we have seen that all these substances, they are not deposited in the peripheral layers, rather they are deposited in the inner layers. So again, this statement is also wrong. Then presence of parenchyma cells, functionally active xylem elements and essential oil. Again, this is also not the case because we do not see any kind of parenchyma cells or functionally active xylem elements because all these cells, they are present on the epidermal part, not in the central part of the secondary xylem. All right. So with this, option A and B are the correct statement. That means A and B only over here. Option A is the correct answer. Clear? Now, question number 34. Now, question number 34 says that the appearance of recombination nodules on the homologous chromosomes. All right. Appearance of recombination nodules on the homologous chromosome during meiosis that characterizes. All right. Now, recombination nodules that are being observed in case of the homologous chromosome and that too during the process of meiosis. So this is basically indicative of the process of crossing over. This is a very direct question that has been asked. Because see, if there will be the appearance of recombination, recombination is basically the exchange of the genetic material. It is the physical exchange between the genetic material, or it's a kind of a physical exchange that results into exchange of genetic material between two homologous chromosomes. And if there will be recombination, then definitely there will be crossing over, because without this crossing over, recombination cannot be possible, or the recombinant nodule is not possible. So recombination is the result of crossover, you can say, crossing over, which is the physical association between two homologous chromosomes, which is physical association between two homologous chromosome. That means one chromosome will be coming from the father and one from the mother. That means one paternal and the other maternal chromosome. So it is the physical association between two homologous chromosomes, thereby exchanging genetic material. Thereby exchanging genetic material. So once the genetic material is exchanged, so that process is considered to be as recombination. All right, so that is the case. So this is one of the stage of your pecitine. This is the pecitine stage. That means the third stage of the prophase 1 of meiosis 1. All right. So pecitine stage of meiosis 1, you can say. Pecitine stage of meiosis 1 is characterized by appearance of, so this is basically characterized by appearance of recombination nodules. Because one, these nodules can only be formed after crossing over, and crossing over occurs in pecitine only. That's why. Combination nodules. All right. So that means option C is the correct answer over here. All right. Bivalent and synaptonymal complex. This cannot be done. This won't result into, because this is the bivalent formation occurs in the zygotine stage of meiosis 1. This occurs in zygotine, and terminalization basically involves the terminalization of chiasma. All right, and terminalization of chiasma is basically the process of diakinesis. So this occurs in diakinesis. All right, so that's why option C is the correct answer. Now, question number 35. Question number 35 says that given below are the two statements that has been given, and you have to go through the two statement, and in the light of the above statements, you have to choose the correct answer from the options given below. All right. Now, let us take a look. Statement 1 says that Mendel studied 7 pairs of contrasting traits in the pea plants and proposed the laws of inheritance. So definitely this is a true statement, because Mendel was the only one who is considered to be the father of genetics, and he was the only one who studied the contrasting traits in pea plant. That means 7 different pairs. All right. So that means total traits were 14. So statement 1 is actually correct. Statement 1 is correct, because Gregor John Mendel studied laws of inheritance on pea plant. on pea plant with its 7 pairs of contrasting traits. Contrasting traits. 7 pairs basically means 14 in number. So there will be total 14 characters. All right, so this is a true statement. Now the second statement says that 7 characters examined by Mendel by his experiment on pea plants were seed shape and color, flower color, pod shape, color, flower position, and stem height. Now let us take a look. I'll just let you people know that whether this statement is true or false. So all those 7 characters that were taken by Mendel actually. All right. So those were, we can just draw a flowchart, trait or character. Then here will be the dominant trait. And here will be the recessive trait in contrast to the dominant trait that has been taken. So the first character was stem height. Stem height can either be tall, that means P height, tall, and here will be dwarf. The second character was flower color. So flower color is here in case of dominant, it is violet and recessive is white. Then flower position. Flower position, the dominant flower position was axial and the recessive one was terminal. So axial and terminal. Then pod color. Pod color primarily is green and here will be yellow, recessive one. Then pod shape. Pod shape was inflated. Here in case of dominant trait was inflated and here was constricted. Then seed shape. Seed shape was round. The dominant one was round and here will be wrinkled. And then last one was, 7th trait was seed color. So seed color in case of dominant was yellow and the recessive was green over here. All right, so these were the traits, 7 different traits. Now let us take a look. So it was seed shape and color, true. Flower color, true. Pod shape and color, true. Flower position and stem height, true. So that means statement 1 and 2 both are considered to be correct over here. All right, so we can say that the statement 2 is correct. Option A is the correct answer. Now let's talk about question number 36. So this question says that transposons can be used during which one of the following processes? All right, so there are certain processes mentioned below in the form of options. So you have to identify the transposons can be used during which of these options, whether it's polymerase chain reaction, PCR, or gene silencing, or autoradiography, or gene sequencing. All right, now let me tell you that basically these transposons are considered to be as the mobile genetic element. All right, let me tell you about transposons first. So these transposons, these are mobile genetic elements. These are mobile genetic elements, or which are also considered to be as jumping genes. that has the ability of moving from one place to the another place, or from one side to the another side in a DNA sequence. All right. So basically, since these are the mobile genetic elements, also called as jumping genes, so the first option which is given over here, that is polymerase chain reaction, PCR. Now PCR is one such reaction where the DNA copy number is being increased. All right, so this is one such reaction that does not requires the involvement of transposons. So definitely option A is not the correct answer, because polymerase chain reaction, which is PCR. All right, so PCR is used to increase the copy number of DNA. Copy number of DNA. And that's why this does not involves or does not need transposons. And does not need transposons. All right. So definitely this is not the correct transposons actually. Transposons. So definitely this is not the correct answer. Then there is gene silencing. Gene silencing can be considered to be as an option, but let us take a look into the other options as well. So the third option is autoradiography. Now autoradiography is basically one such method that involves the process of hybridization. And this is one such technique which is used for a kind of it is one such technique which is primarily being used for detection of for a particular any DNA segment or for any particular RNA. So definitely this also not doesn't involves any kind of involvement of transposons, because autoradiography usually follows the method of hybridization. And since it follows hybridization method, so that has nothing to do with the transposons. Again, this is not also an option. Then the last option is gene sequencing. Again, gene sequencing is also one such method in which the transposons are not at all required, or you people might also not have heard regarding the transposons or these jumping genes in case of the gene sequencing. So D also does not involve, that means gene sequencing that does not also involves transposons. So gene sequencing does not involves transposons. So the only correct option that is left is the option number B over here, which is your gene silencing. Because in case of the gene silencing, these transposons they can be considered they can be considered to be the source of the complementary RNA for the process of RNA inhibition, and that's how they can be used it. So we can say that the B option is that transposons can be the source of complementary RNA. So this can be the source of complementary RNA. Just a second. So this can be the source of complementary RNA for RNAi method. All right. So which is used to replicate, that replicates, since this is a jumping gene, so this can replicate via an RNA intermediate. All right, so that's why the correct answer here will be gene silencer. That means option number B over here. All right, now next question is question number 37. Now question number 37 says that while explaining the interspecific interaction of the population, plus sign is assigned for the beneficial interaction, minus sign is assigned for the detrimental interaction, and 0 is assigned for neutral interaction. So this we already know about it. So which of the following interaction can be assigned plus for one species, for one species, and the other for the another species involved in the reaction, or involved in the interaction, you can say. All right, now let me just tell you that let us start with the very first interaction, which is predation. Now predation is one such interaction where the predator is benefited, so this is benefited, and the prey species actually, and the prey species is harmed, and the prey is harmed. That means plus and minus interaction. So the first option actually has turned out to be the correct option, but let us take a look into the other options as well. While amensalism is one such interaction, amensalism, in which one species is harmed, that means minus, and other species is neither benefited nor harmed, is neither benefited nor harmed. So basically we can say 0. So this is a kind of a minus and 0 interaction. So this is not the case. Then option C is commensalism. So commensalism is again one such interaction in which one species is benefited, and since the species will be benefited, so this will be a positive thing for the species, and the other species, again, other species is neither benefited nor harmed. So this is a 0. That means neutral interaction, All right. While the D option is competition. Now competition again is one such interaction in which both the partners involved are basically harmed. Competing species, both the competing species involved are harmed. So both species are harmed. Both species are harmed. That is minus minus. All right. So with this, we have come to know that option A is the correct answer, which is predation. All right. Now, question number 38. So question number 38 says that which part of the fruit labeled in the given figure makes it a false fruit? All right, so this is a figurative question that has been asked from your second chapter of 12th, which is sexual reproduction in flowering plants. All right, so again, this is a kind of a direct question that has been asked from the figure itself. Now, let me tell you that what is a false fruit. So we know that false fruits are those fruits that develops from parts other than ovary. All right, and it is usually developed from thalamus. So basically, false fruits are the fruits that develops from parts other than ovary, like thalamus. Thalamus. So thalamus is the main part that contributes in the formation of the false fruit. And the example of false fruit usually includes apple, then cashew nut, strawberries. All these are the examples. All right. So we just have to look into the figure regarding the thalamus part. So if you can see, A is the mesocarp, then B is the endocarp over here, then C is the thalamus, and D is the seed. So the direct statement has been, option has been given. So it is option number C, that is thalamus, is the correct answer. All right. Now, question number 39. Now, question number 39 says that match the plant with the kind of the life cycle it is going to exhibit. All right. So you have to match the column with regarding to the life cycles being found. All right. Now, the first one is the Spirogyra. The first one is Spirogyra. Now, let me tell you that Spirogyra is a green alga. All right. So this is basically a category of green alga. And alga, if you people know that alga, basically that means algae usually have a haplontic life cycle. That means most of the algae, they usually have a haplontic life cycle, except few of the exceptions are also there. So let us take a look that the main life cycle is haplontic only. So you can see that dominant diploid sporophyte vascular plant, no. Then dominant haploid free living gametophyte, this can be the option. Dominant diploid sporophyte, no. Dominant leafy gametophyte, no, not at all. So that means this is the correct option over here. This is the A1. So this says that it has haplontic life cycle. Haplontic basically represents to gametophyte only, free living gametophyte only. So that's why. That means the main dominant phase is the haploid phase, gametophytic phase. So it has haplontic life cycle. Clear? Now the second one is fern. And fern is a category of pteridophyte. That includes horsetails and ferns, if you people remember it. And in case of pteridophyte, the main phase that is being included is basically the dominant phase of the pteridophyte is usually the sporophyte, which is a diploid phase. So dominant diploid phase, and they have a reduced thallus, and their thallus, however, their thallus is there, but their thallus is still independent, and that thallus is referred to as the prothallus, which is being represented in the form of gametophyte. So basically, main dominant phase is sporophyte, which is diploid. All right. While gametophyte is represented by prothallus. While gametophyte is represented by prothallus. So with this, if we just come to know, then fern, it will belong to option number 3 over here, this one. This is the B option over here. All right. Then there is funaria. Funaria is a moss category of bryophyte. All right. And in case of bryophyte, this is just the opposite condition of the pteridophyte. All right. So funaria, which is a moss, that means bryophyte. Dominant phase over here is gametophyte. So dominant phase is gametophyte. All right. And in this case, in this category, the gametophyte is usually leafy. It has a leafy gametophore. So we can say the gametophyte is of leafy category. All right. And since the sporophyte is dependent on the gametophyte, so that's why we can say that while sporophyte is dependent. Is dependent on gametophyte. So of course, this is the case. Now let us take a look. So two options are left. Dominant diploid sporophyte vascular plant, no. Funera is going to match up with dominant haploid leafy gametophyte, alternatingly with partially dependent multicellular sporophyte. So this is C option over here. The last is the cycas. And cycas we all know is a category of gymnosperm. It's a gymnospermic species. All right, where if we just know about cycas, which is a gymnosperm. So in case of gymnosperm, again, gymnosperm, and in case of angiosperm also, the main plant body is sporophyte itself. So dominant plant is sporophyte. Is sporophyte, while gametophyte is actually reduced. Gametophyte is reduced. All right, so that is the case. So this is going to match up with the only left option, which is this one. Now let us just match the following options. So A is going to match up with second. A is going to match up with second, okay. Then B is going to match up with third one. B is going to match up with third one, this one. Then C is going to match up with fourth, and D is going to match up with one. So that means option B is the correct answer. All right, now question number 40. Now question number 40 says that you have to match the column with the respective forms. All right, now list 1 and list 2 has been given. Now let's talk about the different chromosomes. So this is basically the different categories of chromosomes that has been enlisted on the basis of the position of the centromere. So metacentric chromosomes are those chromosomes. Metacentric chromosomes. So basically metacentric chromosomes are those chromosomes who have a centrally localized centromere, and both arms are equal. It is actually placed at the center. The centromere is situated at the center. So if I just draw it, so this will give an appearance like this. This is the centromere, and this is going to give an appearance like this. So this is a metacentric chromosome. So the centromere in the center. Centromere at center of chromosome. It is at center of chromosome. Then there is acrocentric chromosome. So A is going to match up with centromere in the middle, forming two equal arms. Definitely. So this is the correct answer. Then acrocentric chromosome. Now acrocentric chromosomes are those chromosomes in which one chromosome, one end is very short, one arm of the chromosome is very short, and one is very long. So that is called acrocentric chromosome. So it can be drawn like this. One arm is very short, and then the other arm is very long like this. So this is the acrocentric chromosome. That means basically situated one extremely short arm and the one extremely long arm. You can see centromere situated close to the end. So this is the short arm, very short arm you can say, and this is the long arm, very long. All right, so definitely this is the correct answer, B. Then submetacentric chromosome. Now submetacentric chromosomes are those chromosomes where the centromere is slightly situated or slightly just deterred from the center of the chromosome. So submetacentric chromosome. So this will give an appearance like this. Like this. All right. So slightly centromere, slightly away from the center. Centromere, slightly away from center. So centromere slightly away from the middle, forming one shorter arm and one long. So this is the C option. And the last option which is left is the telocentric chromosome that has only one terminal left. This one. That means it can be drawn like this. Telocentric. Chromosome. So this is having an appearance like this. Terminal end position, you can say. There like this, but actually at the top. Or we can do it like this. And to get an appearance like this. Actually at the terminal. So that is the case. All right. So let us match up with the different options. So A is going to match up with third. Then B is going to match up with first. Then C is going to match up with fourth. And D is going to match up with second. That means option 1 is the correct answer. Question number 41. So question number 41 says that addition of more solutes in a given solution will raise its water potential, lower its water potential, make its water potential 0, or not affect the water potential at all. Now let me tell you that solutes, all right, addition of solute or solutes or the presence of solutes in a solution is called as solid potential. All right. So we can say that, just a second. Addition of more solutes in a solution. Now, when more solutes will be added in a solution, so this will make the solution, this will actually make the solution or the solute potential of that particular solution to be more negative. All right. So this will make solute potential of the solution. Remember, solute potential I'm talking about, which is always negative. Solute potential is always negative, which is psi p, sorry, psi s. All right. So this will make the solute potential of the solution to be more negative. Remember, it will make more negative. All right. And if there will be more negative kind of a solute potential that is developed in case of the solution, so definitely the availability of the free water molecules will be very, very less. And you can say that, and so the availability of free water molecules will be very less. So this will indicate that the water potential is actually going to be lower. All right. This means water potential will be lower. Water potential will be lower. All right, so that is the case. Now let us see what are the statements that has been given. Now addition of more solutes in a given solution will raise its water potential, not at all. This is going to decrease the water potential, not it will raise it. All right, lower its water potential, yes. This is going to lower its water potential because since more amount of solute will be added, so there will be more amount of solutes that will be present in the solution, and as a result the solute potential will be more and more negative, and so the availability of the free water molecules will be very less. And since the free water molecules will be less, definitely the water potential will be going to be lower. All right, it makes the water potential zero. Again, this is also not an option because the water potential is zero, or it is considered to be as the highest when it is pure. All right, when it is not having any kind of dissolution of solutes, so then only the water potential can be zero. That means pure water usually has a water potential of zero, so definitely this is not a pure water because here it is asking about solution. All right, now it doesn't affect the water potential at all. Again, this is also not an option because definitely water potential will be affected if a single amount is added or whether it's a ample amount of solutes is added. So definitely water potential will be affected. So the option B is the correct answer. Now, question number 42. Now, question number 42 says that which of the following occurs due to the presence of autosome linked dominant trait? Now, it is asking here about the autosomal linked dominant trait. Now, out of all the traits, now let us just classify that which are, dominant and which are recessive, or which are, sex linked. So basically, sickle cell anemia. All right, sickle cell anemia is an example of autosomal recessive trait. Sickle cell anemia, this is an autosomal recessive disorder. Autosomal recessive disorder. All right, then there is hemophilia and thalassemia. Now hemophilia is an X-linked recessive disorder. Let me tell you that hemophilia, this is an X-linked recessive disorder. Recessive disorder. Then there is thalassemia. Now thalassemia is one such disorder which is again autosomal recessive. Thalassemia. An autosomal recessive disorder. The only option left here is the one option which is myotonic dystrophy. This is an autosomal dominant disorder because it occurs because of the dominancy of the autosomal linked dominant trait. All right, so myotonic dystrophy. This is autosomal dominant disorder. All right. So the correct answer is option D. Now, question number 43. Now, question number 43 says that which one of the following will accelerate the phosphorus cycle? All right. So there are two important cycles that are there in your course that you people might have studied it: nitrogen and phosphorus cycle. So phosphorus cycle, burning of fossil fuels, the option givens are burning of fossil fuels, then volcanic activity, then weathering of rocks, and rainfall and storms. All right, now let me tell you that phosphorus cycle basically represents an example. Basically, there are two types of cycle. All right. So one is having atmosphere as the reservoir pool, and the other is basically having, one is actually called as the gaseous cycle, and the other is called as sedimentary cycle. So phosphorus cycle comes under sedimentary cycle actually. Phosphorus cycle is a sedimentary cycle. That means it is not, just a second, sedimentary. So this means that it is not having any gaseous, output. So it is not having any kind of gaseous output, you can say. So it's a sedimentary cycle. All right. And the reservoir pool of this phosphorus cycle is considered to be as the Earth's crust. All right. So the reservoir pool for this cycle is Earth's crust. Because once the weathering processes of rocks is being done, whatever the rocks that contains phosphorus in the form of phosphate, all right, so those are dissolved in the soil. So basically this Earth's crust, or you can say weathering of rocks, this releases phosphorus in the form of phosphate. Releases phosphorus in form of phosphate. All right. And that's how they are being absorbed by the thin layer of soil. Then they form a thin layer of soil, and then, the different succession plans are being taken into account. So burning of fossil fuel, again, this has a gaseous reservoir, so definitely this is not an option. Volcanic activity, not an option. Rainfall and storms, again, rainfall and storms is a category of carbon cycle and oxygen cycle, but not the phosphorus cycle. So definitely weathering of rocks is the correct answer. Option C. Now, question number 44 over here. Now, in the following palindromic base sequences of DNA, which one can be cut easily by a particular restriction enzyme? So, which can be cut easily into a particular restriction enzyme? If you will see, palindromic base sequences are those sequences which you either read from the left side or from the right side, that is going to read the same. Like words like Malayalam and all those examples have been given in your textbook also. Now, out of the four options that we have seen, the best example that has been given, so this is also a direct question that has been asked, because restriction enzymes, they recognizes a specific site in the DNA sequence. And the site for the palindromic base sequence that has been given, if you just look into the different options, then the correct answer here is actually the sequence belonging to 5' G double A double T C 3', and then 3' C double T A G over here, 5'. All right. Now the cut where they are being used, that means one can easily cut by the particular restriction enzyme. So the cut where they are being marked, so the cut is over here. That is between A and G. So restriction enzyme, and this is an example of eco-RI I'm talking about basically. So restriction enzyme, which is this one, is used to cut between base pair A and G. Remember, this is basically used to cut the base pairs between A and G so that the orientation will be same. All right, orientation will turn out to be same if you read it from the left side or if you read it from the right side. So if I just cut it out, so how the sequence goes on, so this will be like this. This will be 5 prime, then G will be here, and then AA, TTC will be here. And then 3 prime over here, then C, TT, AA will be here, and then G will be here. So this is how the sticky ends are created and then they are joined up. You can see. All right. So these are the sticky ends which are created over here. All right. So that's why the correct answer here, if you will see it, so option B is the correct answer for you. All right. Now, question number 45. Now, question number 45 says that you have to read the following statements on lipids and find out the correct set of statements over here. All right. So lipids, the statements on lipids has been given and you have to find out the correct statement. Now, the first statement says that lecithin, which is found in the plasma membrane, is basically a glycolipid. All right. Then lecithin, now let me tell you that lecithin, which is found in case of the plasma membrane, that is not a glycolipid. This is actually a phospholipid. So lecithin, and this is the most common type of phospholipid which is being found in the plasma membrane of animals. So lecithin is a phospholipid found in plasma membrane. So definitely this is a wrong statement. All right, this one is a wrong statement over here. Now, the second statement says that saturated fatty acids possesses one or more carbon-carbon double bonds. Now, again, this is a wrong statement because it is not the saturated fatty acid that possesses it, but it is the unsaturated fatty acids. All right, because saturated fatty acids do not have any kind of double or triple bonds. So this is also a wrong statement because saturated fatty acids do not have double or triple bonds. All right. So this is again a wrong statement. Then the third statement which says that gingerly oil has lower melting point, hence remains as oil in winter. All right. So generally, since oils usually have a lower melting point, all right, so that's why they remain oil in winter. So this is a probably correct statement. All right. So since oils have lower melting point, so since oils they have low melting point, that's why they are oil in winter, or they remain as oil in winter rather. All right, so this is the correct statement. Then option D says that lipids are generally insoluble in water but soluble in some organic solvents. This is also true. Lipids are generally insoluble in water, all right, but since they have the presence of those uh, hydrophobic tails and hydrophilic heads and all those compositions, so they are insoluble in water but soluble in organic solvents like ethanol or something. So this is a true statement. And the next statement says that when fatty acid is esterified with glycerol, monoglycerides are formed. Again, this is also a true statement. This is also a correct statement that when the esterification of fatty acid is done with glycerol, the monoglycerides are being formed. All right. So that means out of 5 statements, options C, D, and E are correct. All right. So let us take a look into the statement. A, B, C, then option C, C, D, and E only. They are considered to be as the correct statement, correct option. All right. Now, question number 46 says that what is the role of large vascular bundle cells found around the vascular bundles in the C4 plants? This is basically asking about the role of the bundle sheath cells that is found around the vascular bundles. So, basically, what is the organization? So, this something looks like this. This is a pictorial representation that I'm drawing. So, this is a bundle sheath cells are actually very thick cells that are found around the vascular bundles like this, enclosing the vascular tissues like this. All right. And if you just, there's a layer of gadolinium over here. And the upper part basically is the portion of the xylem. This one. And this is the floor. This is the upper part is basically the phloem part, sorry. And the lower part can be the xylem part like this. Like this. All right. So this is the phloem. This is the xylem. This is the bundle sheath cells, which are actually very, very thick. And these bundle sheath cells, these are the peculiar feature of C4 plant only. This is not found in any other categories of in the leaves or in the mesophyll cells, you can say rather, of the C4 plants, not in any other categories. So these bundle sheath cells, the large cells, the main function of these bundle sheath cells, the options given over here is to provide the site for the photorespiratory pathway. No, this is not the case, because see, bundle sheath cells are those cells which plays a very important role in not escaping out. It doesn't allows the CO2 which is liberated during the process of oxidative decarboxylation. So it doesn't allows the CO2 to escape from the cell. All right. So it basically entraps it. All right. So that's why. So it doesn't allows CO2 to escape. And when the CO2 won't be able to escape, the CO2 will actually, this will actually be fixed by the Rubisco enzyme in the presence of RUBP to give rise to carbohydrate or food. All right. So does not allows CO2 to escape, and so it is fixed in presence of RUBP and Rubisco to form sugar or carbohydrate, you can say, as a product. As a product. And this is in case of, that means they will be entering into Kelvin cycle. So to increase the number of chloroplast for the operation of the Kelvin cycle, this can also be the option. This is actually a true statement because these bundle sheet cells, they have greater number of chloroplast as compared to any other cells. So this can be the correct statement. All right, because these cells, since these cells are large, so they have larger number of chloroplasts to perform the Kelvin cycle. All right. So this is the true statement, but let us take a look into the other statement as well. To enable the plant to tolerate high temperature, basically this cannot be considered. See, larger bundle sheath cells are found in case of the C4 plants. All right, but this has nothing to do with toleration of the higher temperature. Those C4 plants, they tolerate high temperature, they have higher productivity as well, but it has nothing to do with this part. So definitely this is not an option, appropriate answer. Now, to protect the vascular tissues from highlight intensity, no. Again, this is also not an option. So the correct answer here will be option number 2. Now, question number 47. Now, question number 47 says that if a geneticist uses the blind approach for sequencing the whole genome of an organism, followed by assignment of function to different segments. That means the whole genome is being sequenced, blind approach is being done, and the whole genome is sequenced, and you have to assign it different segments, and you have to assign the different segments that are there. So what will be the methodology adopted by him? That will be called. So this is, you can say that this is a very, you can say this is an easy kind of a question that has been asked actually, because see, if you have to assign the functioning of different segments, then definitely you have to know the segments. Definitely it will be very, very required to know each of the to know each of the segments. So for that purpose, sequencing is very, very important. That means gene sequencing is very, very important. All right, because it contains all the coding genes and all the non-coding sequences, and later on, depending upon the categories of the coding and non-coding sequences, you can assign the different functions as well. All right, so we can say that sequencing is a process of finding out the coding and non-coding genes. In a whole set of genome, you can say. So in a whole set of genome, it is very, very important that you have to find out the coding gene as well as the non-coding genes. And once you find out this coding and non-coding genes using this sequencing process, and then later assigning, that means the process, this process will be called as, up till this, it will be called as sequencing process. But once you will assign the functions to different segments, that will be called as annotation, because you are assigning specific sequence to specific segments of the DNA. That's why. And process of assigning. So process of assigning different regions in the sequence process of assigning the different regions in the sequence with functions, along with their functions also. So with functions is called, that is considered to be as sequence annotation. That is called sequence annotation. Simple. All right. So that means out of all the options, sequence annotation is the correct answer. Clear? Now question number 48. Now question number 48 says that given below are the two statements that has been given, and you have to read out the assertion and reason. That means it is given in the form of assertion and the other is being given in the form of reason. So let us just find out that whether the reason is correct for assertion or not, or whether those statements are correct or not. All right. So assertion is that Mendel's laws of independent assortment does not hold good for the genes that are located closely on the same chromosomes. All right. So, see, Mendel's law of independent assortment is basically related with the dihybrid clause. All right. This we all know it, and this is one of the laws of important laws of the Mendel's law of inheritance. And this is actually a true statement because Mendel law of independent assortment doesn't holds good for those genes which are closely related on the same chromosome, that is linked genes. It doesn't holds good. So that means assertion is correct. Assertion, that means A, is correct because Mendel's law of independent assortment does not hold true for linked genes. Remember that means the genes which are closely located on the same chromosome. So genes that are closely located on same chromosome. Genes are closely located on same chromosome. So this is actually true. That means assertion is correct. All right. Now, closely located genes assort independently again. So closely related genes does not assort independently because they are the linked genes. So they won't be segregating. Closely linked genes means the linked genes it is talking about. So definitely the reason is basically considered to be as not the correct statement, you can say. So closely linked genes assort independently. No, this statement is actually false. The reason, and because the reason I have already given over here. So the reason is incorrect actually. Reason is incorrect. Because Mendel's law of inheritance, it actually holds good for, those genes which are differently located. That means the genes which are located on different chromosomes. That's why. All right. So you can say that if you just look into the options, A is correct, but R is not correct. That means option C is the correct answer. All right. Question number 49. Now, question number 49 says that the anatomy of springwood shows some peculiar features. All right. Anatomy of springwood shows some peculiar features. Now, you have to identify the correct set of statements about the springwood. So, basically, there are two types of wood. One is considered to be as the springwood and the other is considered to be as the autumn wood. So, springwood, basically, two types of woods are being found. Two types of woods are there. All right, springwood and autumn wood. Basically, these woods represents the secondary xylem only. So one is called as the springwood, and the other is considered to be as autumn wood. Now, springwood has a very active cambium. This wood is called as springwood because it produces greater number of xylary elements because it has very active cambium. So active cambium So since they have active cambium, so they produces greater number of xylary elements. Produces greater amount of secondary xylem. And this is during spring season only actually. But it is just the opposite in case of autumn wood, because in case of autumn wood, the cambium is actually little, it is actually not active, we can say, but it is less active. This is less active. And these xylem, these secondary xylem, they have greater vessels, or they have bigger vessels rather. So these xylem have bigger vessels. So they have bigger vessels also. And it is also having, this and the importance of these xylem is that it is also light in color and having lower density as well. So it is light in color with lower density. That is one of the also important features. All right, now let us take a look that what are the options that has been given over here. So since this is here talking about spring wood, so we will primarily focus on the spring wood also, itself. So spring wood is also called as early wood. Yes. This is also one of the important statement, because spring wood is also considered to be as early wood, while autumn wood is considered to be as late wood. This is called as late wood. So definitely this is a true statement. Then in spring season, cambium produces xylem elements with narrow vessels. No, not narrow but broader vessels, as I told you. So this is a wrong statement. It is lighter in color. Yes. Then the spring wood along with the autumn wood shows alternate concentric rings forming annual rings. True, very true. Because both of them, they constitutes, all right, both of them will combine. That means spring wood and autumn wood, they combine to give rise to autumn, sorry, autumn wood. They combine to give rise to annual rings. All right, so this is the case. Clear? It is having lower density. Yes, this is also true. So this means that option A, C, D, and E are correct. So it's A, C, D, and E. Option B is the correct answer. All right, now question number 50. Question number 50 says that the entire fleet of buses in Delhi, they were converted to CNG from diesel. All buses have been converted to CNG from diesel. Now, in reference to this, which one of the following statement is false? Now, you have to identify the false statement actually. So, CNG burns more efficiently than diesel, it's talking about. Then the same diesel engine is used in CNG buses, making the cost of conversion low. It is cheaper than diesel and it cannot be adulterated like diesel. All right. So basically, one of the most important thing now, let me tell you that here you have to identify the false statement. All right, remember, you have to identify the false statement. Now CNG has some quality. CNG has some very very important qualities over the petrol and diesel, and those qualities are, or advantages of CNG are, basically advantages of CNG over petrol or diesel. The advantages over of CNG over petrol and diesel is that the very first important advantage is that CNG burns very efficiently. It burns more efficiently than diesel or petrol. So CNG burns more efficiently than petrol or diesel. This is a true fact, actually. All right. Then the second advantage is that unlike petrol and diesel that can be adulterated, this CNG cannot be adulterated. Any kind of mixing things cannot be done. All right. So definitely this cannot be adulterated. It cannot be adulterated. The second important thing. And the third important point, which is very, very important, is that it is actually cheaper than diesel and petrol. So it is cheaper than diesel. It's actually cheaper than diesel and petrol. All right. And if you just look into the different statements, so this statement is true. All right. Then this is also true. And this is also true. But the one statement which is left over here is that this one, and this is actually false, which says that the same diesel engine is used in CNG buses, making the cost of conversion low. This actually cannot be the case because the same diesel engine cannot be used. It actually cannot be used in CNG buses because in case of the CNG buses, the CNG engines are different and they have a different installation as compared to this one, your diesel engine. So definitely the same diesel engine cannot be used. All right. So that's why this is a false statement. That means since we have to identify the false statement, so this one is the correct answer. Option B is the correct answer over here. All right.